Good afternoon, and welcome to the Data Core Pre-ADRC Meeting Workshop. My name is Amy Young, and I am from the National Alzheimer's Coordinating Center, and we're pleased to have you all here today. I want to go over just a few meeting logistics. The workshop will go from 2 to 5, followed by the updates from the EDC work group from 5 to 6. We will have a few breaks, because that is a long time to be sitting. So you'll have a few breaks in which you can use the restrooms. The restrooms are just around the corner over there. If you brought a water bottle, we also have bottled water out front. You can refill your water bottle. There's a station there. There are also vending machines down the hall. I hear you can just follow the smell of the pizza. So there's some food down there and drinks if you want to get that. What else? Also, if you could silence your phones during the presentations, we'd really appreciate that as well. During the question and answer period, because we will have microphones and Claire and I will be going up and down the aisles, and we can hand you the microphone if you have a question to ask. And then we'll also be taking questions from the online group as well. There's a lot of people online on Zoom, and Yorgos will be fielding those questions. So he will be asking the Q&A questions. Today, I have the pleasure of introducing the chair of the Data Core, who is hosting the meeting, Sudeshna Das. Sudeshna is an assistant professor of neurology at the Harvard Medical School, a member of the staff at Mass General Hospital, and a faculty member at the Harvard Stem Cell Institute. Please join me in welcoming Dr. Sudeshna Das.
Thank you, Amy. So I'd like to welcome everyone to the Data Core pre-meeting workshop today, and welcome to Chicago, sunny and windy. And we have an incredible speaker lineup, and we had a really overwhelming response to our call for proposals. We had like over 50 proposals, and we just had slots for a few of them to talk over here today. So, but welcome everyone else if your talk wasn't selected to do a webinar later on, and NACC does it. We have periodic webinars as well. And couple of announcements other than, silencing your phones and having the mic runs for Q&A. So please silence your phones and you'll hear the session today. I'd like to take this opportunity to thank Northwestern for being our host today, especially Paige and Debbie here, who's the data manager of the Northwestern ADRC, and the fabulous NACC ladies, Amy, Hannah, Claire, and everyone who has made this whole hybrid operations smooth as butter. So welcome everyone. And I'd like to introduce the first speaker. That's Dr. Sheng Luo, he's a new Data Core steering committee member. He's going to be talking about the genetic association between epigenetic aging acceleration and the progression of MCI to AD.
Thank you. Thank you for the introduction. So this work has been published in this journal. So let me see. Can I? Okay, yeah. So okay, so yeah, has been published earlier this year. So now, so a little bit of introduction. So we all know that the aging is one of the leading risk factor for AD. Some previous studies show that the detrimental relationship between the accelerated brain aging and AD early stage neurodegeneration. So here, particularly, we want to look at this epigenetic age acceleration pathway. So this pathway actually indicating the difference between the estimated age by methylation data and your real age. So this EAA pathway is the effective biomarker to predict multiple age-related phenotypes, such as the cancer, obesity, and so on. So now, specifically, we want to look at two EAA measures. IEAA, I stand for intrinsic, and also EEAA, E stand for extrinsic. So now we have seen the inconsistent results on the association of the EAA and dementia risk. So the hypothesis here is that we want to look at the EAA-related genetic variant and see if they are associated with the progression from MCI to AD. So the population here, we used the two of them, ADNI and NACC. So from ADNI, we identify 767 individuals with MCI at baseline, with also available genotype and clinical data. So among them, 294 of them developed dementia during the study. Also, we identify 1,373 subjects from NACC. Among them, 864 developed dementia. So now, if we look at this, the IEAA and EEAA two pathway, we select about, for each of them, 275,000 or 277,000 SNPs with the p-value less than 0.05 and a minor allele frequency larger than 0.05 in a European population. And then we perform imputation because some of them have missing data. So using the regular techniques here, so such as say the MAF larger than 0.01 and so on. So without getting into all this detail. So now the imputed SNPs with the MAF larger than 0.05 and imputation quality score larger than 0.3 was selected for the final analysis. Okay, so that's the busy slide. Now we first run the univariate analysis to test the association between each of the SNP and progression from MCI to AD using Cox model, while adjusting for other covariates such as the age, sex, education, race, and some principal components. And also, most importantly, ApoE2 and 4. We also adopted LD clumping and survival lasso to select SNPs with the independent effect. After that, we construct the polygenic risk score using precise tool and also using the effect size estimated from the previous EAA GWAS study. We also look at the correlation between the PRS and the longitudinal change of some CSF biomarkers and imaging biomarker. And also we look at the functional annotation. So here is the study workflow. So at the beginning, we identify in each of the pathway about 275,000 SNPs. And then in the discovery stage, we use the ADNI study. And then with the criteria of p-value less than 0.05 and then the Bayesian FDP less than 0.8, we identify about 8,000 or 6,000 SNPs. And then we go into the replication stage to validate that in NACC data set. So eventually we identify 70 SNPs from the IEAA pathway and 81 from the EEAA pathway. And then from there, we use the linkage disequilibrium clumping and also lasso regression to identify 22 and 16 SNPs in these two pathways. And from there, we can construct the PRS and look at how the PRS can help us to predict the MCI to dementia conversion. And also we can run the longitudinal analysis. And also we can do the functional annotation. So these two tables that show the demographic variables for ADRC and NACC. I want to skip that. Also, this Manhattan plot show the SNPs we identify using the criteria of p-value less than 0.05 and Bayesian FDP less than 0.8. So from here, we identify 70 SNPs from IEAA and also 81 from EEAA pathway. Oh, sorry. Okay, so we also using survival lasso to narrow down the SNPs we want to look at. So eventually, from both studies, we identified 22 SNPs in IEAA and also in EEAA, we identified 16 SNPs. So we use, so here show the multivariate Cox model result using this 16 SNPs or 22 SNPs. So I want to skip that. So after that, we calculated the polygenic risk score. So here, this the Kaplan-Meier curve show you the how the PRS can distinguish the people with the faster progression versus slower progression. So the yellow line here show the people with this higher PRS, and then the blue line show the people with a smaller PRS. You can see that in each of the graph, it indicates that the people with a higher PRS progress faster into dementia. So in the ADRC, let me see. Okay, yeah, in ADRC study, we can see both of these PRS that have a significant p-value, while in the NACC, this one. Okay, so only the IEAA is not significant, and the EEAA PRS is significant. And also, I think this is one of the key results. So we look at multiple Cox model and see how the PRS can help us to predict. So the first model is the so-called demographic model. We only include age, sex, education, and race. So we have the C statistic of 0.555 is relatively low. Now, if we add the ApoE2 and ApoE4 into the model, the C statistic increased to 0.636 in ADRC. So which is the significant increase. Now, if we add the additional PRS from IEAA, so the C statistic increased to a little bit to 0.641, which is not significant. If we add the EEAA PRS, it also had a very marginal increase, which is not significant either. So similar finding from NACC. So here it showed the longitudinal data analysis to see how each of these longitudinal outcome in cognitive or in CSF or biomarker or imaging biomarker had to do with the PRS. So the key finding here is the IEAA PRS is associated with the CSF A beta trajectory. And also the EEAA PRS is associated with the FUS imaging biomarker. So to wrap up, we identify multiple genetic variant with pleiotropic effect on the EAA pathway. They are associated with the progression from MCI to AD. It suggests shared genetic architecture between these traits. We also did some polygenic risk score analysis. It reviewed the combined effect of those SNPs and AD progression, and also the longitudinal change of A beta in CSF and volume change in FUS gyrus. I skipped the functional annotation results, but however, we don't find any significance between the association of the PRS and AD progression. So that result may suggest that those identified SNPs do not contribute substantially to the effect of the epigenetic changing on AD progression. So in the future, if we adopt some advanced statistical learning approach, so we might identify more genetic variant in those pathway. Hopefully that will improve the performance in the AD risk prediction model. And at the end, I want to acknowledge my my collaborator, Dr. Michael Lutz, who is also the co-lead of Duke ADRC's data management and statistical, and research scientist Hong Liang, and also thank the participant and investigator of ADNI and NACC. Thank you. Yes. Oh, hi, Xiong-Jie. Hey, good to see you.
Can you, wait, microphones, Sarah? Can you hand me the microphone? If you could say your name and where you're from before you.
Sure. King-Jie Xiong from Washington University, St. Louis. So this is really great work, Shane. I know it doesn't seem to show a statistical significance in some of the measures. It could be sample size or those issues, right?
Yeah.
So I wonder why. My impression is go to NA, go to NACC, you're going to have a much bigger sample size than what you have. Are you subsetting some of the subjects, or due to some of the subjects don't have the genetic data?
Yeah, that's exactly right. So yeah, here we select the people with MCI at baseline in both studies.
Right,
so that
right.
Significantly limited our sample size. And also we have to take the people with the available genotype and clinical data.
Right.
So that also reduced our sample size.
Right, right. You didn't select by race, right? All the different races
Right.
are included.
Right. Yeah.
All right. I think that's probably the sample size you end up with. Now, another question is people die, especially those with MCI, they're going to die before they develop AD. So is the Cox model somehow handling this, or?
No, that is the limitation. Yeah, sure. It's more like a competing risk issue.
Right,
So,
right,
yeah.
right.
So
right.
Yeah,
Right.
it can be accounted for. Yeah.
So the PRS, that score is based on 22 SNPs.
Yeah, one is 22. The other one I think is 17. I think 16. Yeah.
Great,
Yeah.
great. Thank you. That's really nice work. Yeah.
Thank you for your question.
Thank you, Shang. That's a really nice question. And that's a wonderful study. And my question is like, how did you select the first set of 275 and 77,000 SNPs? And are any of them like are in the LD with ApoE? Because ApoE obviously is a big genetic factor, a genetic risk factor for AD. And if you were to include ApoE, your model, results change. So did you verify that those SNPs were not in LD with ApoE?
Yeah, thank you for the question. So the first, to answer your first question, how did I select these the 275,000? So those are the SNPs that are located in those pathway. So those are the SNP in the pathway, in those two pathways. So we could identify them. Yes. Yeah. Yeah, and then to answer your second question, so yes, we did check the linkage disequilibrium and make sure we don't include those SNPs that are in close LD with ApoE. Yeah.
Thank you.
Thank you.
We have a question from whether the Cox model accounts for left censoring.
No. Yes, thank you. Yeah, there's another limitation. Yes. Thank you.
Thank you, Sheng. I'd like to welcome our next speaker, Dr. Chad Murchison from the UAB Exploratory ADRC. And he's going to be talking about data-driven development of the UAB Exploratory ADRC.
Thanks, Adeshna. Well, I just kind of scoot ahead here. I do want to mention, if you look at the agenda for the workshop today, you're going to see a lot of very excellent and very elegant uses of center data to build these very sophisticated models that are super exciting. But I think it's important to remember that when we think about the data cores of the ADRCs, even though the statistical analyses may be like the big rock stars that we're all really kind of interested in, there's another aspect, because we're really kind of wedging two things under a trench coat for a data core. And the other aspect is really kind of data management. And that's what I really want to talk about today. In particular, how we as an exploratory center, one of the 4 centers like Cleveland and Nevada and New Mexico and Vanderbilt, are really trying to grow into one of the fully fledged ADRCs and the way that we're using our data in order to kind of help promote that process. So I'm just going to talk a bit about the background of who we are, and then kind of go into how we're operationalizing our data for process improvement and where we kind of plan to go from here. And I do just want to say I am going to be showing a lot of applications and reports that I've built in the process. And this is using data that has been generated or otherwise randomly anonymized because PHI. So who is UAB? So we are in fact an ADRC. We are official, exploratory or not. And like all the other centers, we do have a thematic focus, and ours in particular is on deep South disparities in Alzheimer's. In particular, we are the US region with the largest population identifying as Black or African American. And in addition, a number of risk factors that are commonly associated with AD. So the usual suspects like diabetes, cerebrovascular disease, and obesity are especially prevalent in the area. So we have this intersection of race and region, all of which is influencing Alzheimer's. So we began our pilot phase in 2018 with some great support from the university, as well as some angel funding. And we were funded as a fully fledged P20 exploratory center in September of 2022. What an exciting time to be alive, to be like rolling these things out. And we have 2 pre-specified cores. We have our clinical core, like all the centers do, and we have our CODI core, which is our community outreach, diversity, and inclusion. And even though these are our 2 official cores, we still are building up the architecture and the infrastructure to be able to show the capacity of a full P30 funded center. So that includes things like biomarkers, neuropathology, imaging, and data, which is where I kind of come in. So our goal here has been to really build a longitudinal clinical cohort that is being dictated by the requirements of NACC, but is also aligned with what we want to be able to do at UAB. So we are collecting our clinical data via the UDS, and we use the same kind of workflow that a lot of other centers do, where we collect the data on paper forms, but we do want to make sure that we have an emphasis on electronic capture wherever possible, and really kind of limiting things like transcriptional or annotation errors, things of that sort, really trying to leverage QA/QC where we can. Our neuroimaging and our neuropathology are still very nascent, but maturing as we kind of go through. They do things on their end, and we are facilitating this cross-core collaboration to make sure that everybody is getting the data that they need. In particular, we are utilizing REDCap for data entry alongside processing using R, which is my tool of choice. If data is the paint, then R is my canvas. And we're really using long-term persistent storage in a true formal relational database, since REDCap has many strengths, but data storage is not really one of them. As we go through this, we do leverage other resources as dictated by need, because you want to use the best tool for the job. So how have we been operationalizing our data in order to kind of improve the process here at UAB? Basically, we're really trying to manage the conflict on 2 fronts, between data collection and really trying to make the experience for the participants as easy as possible, and then trying to improve the process and really kind of make sure that we can do what we need to do as a center. And in this respect, we really have 2 large goals for data utilization. We want to refine our study activities for the clinicians and the raters and the rest of the center staff. And we also want to leverage that data in order to improve our operations as a center as a whole. In the act of doing this, I've built many applications, reports, and other documentation that has been tailored to UAB. I'm going to talk a bit about a dashboard that I've developed that has really been beneficial on a number of fronts, as well as some of the other reports and tools that have been requested to me from our staff based off what they found, as well as ways that I've tried to improve electronic data capture via REDCap, which we'll go into significant detail later on with the work group discussion. And again, all this is building towards the goal of being a P30 center and really kind of establishing us as a true collaborative site. So anyone who has actually worked with REDCap output or tried to fill in some of these paper forms that we have as part of the UDS packet knows that they can be a little cumbersome to work through, especially when your clinicians are trying to do diagnoses. One of the first things that were requested of me was to make it just easier to work with. And so we can see here some examples of the CDR. And it can be a little challenging to work through this, kind of figure out, well, what's good, what's bad, like yes and no mean different things depending on the context of the question, whether it's coming from the collateral or the subject. How can we really kind of improve that? So one of the first things that I did was develop like markdown reports that are much more direct and intuitive than what you would generally see from just kind of like the raw output that we have from the data entry steps. In addition, I also put together a template that takes a lot of the neuropsych scores that we get not only from the C2 forms, but also the stuff that we collect on the side in order to be able to do calculations for Z scores that are adjusted for age, education, sex, and race, and then can map these to percentiles that we can then use to indicate where a given subject is actually deficient for a given domain, whether it's attention, memory, language. And this can really kind of help as our clinicians are filling out the D1 form and trying to figure out what the actual etiology of the disease really is. We can combine these things into reports in nice little like concise packets that are much more digestible for our clinicians as they go through the review and conference process. And we can actually distribute these using REDCap. We can use combination of surveys and then hook files in order to easily send these where they need to be. And we can do all this with very little direct data entry by making use of like R scripts that I developed or APIs. So really kind of get everything together and use the electronics where we can. In addition, I mentioned some of the improvements that we wanted to do with REDCap in particular. So again, we've had some great dictionaries thanks to people like Kansas, who have been instrumental with many ADRCs, not just us. And we've really tried to make improvements, though, to better kind of leverage stuff longitudinally. You can see here some examples of what I've done where we can take, there we go, hey, it's working, where an initial visit has data filled in and then gets piped into a report, which can then be used to actually kind of inform follow-up questions. So we can see transitions for, say, the ability to live independently from one year to the next. And this is really kind of useful, not only in terms of validation and QA/QC, but it also helps engage in conversations with the participant, which can actually help the process as a whole and that subject experience I mentioned. In addition, I've developed other reports to kind of help with processes. We have things like up there at the top, identifying individuals who are coming due at the 11th month mark for a follow-up visit so that we can actually have our clinical core really kind of facilitate scheduling. And then we know in terms of the uploads that are required for UDS for NACC, we need to make sure that we have a complete packet, that it's not an error, and we can actually leverage the data that we're collecting and REDCap in our database and make sure everything is actually kind of filled in before we go through the processing steps. In addition, I put together a little scheduling app that again is directly connecting to the REDCap via API in order to pull dates that are important and actually build out a timeline for a given set of visits. So we can do things like take the contact date, the enrollment date, and the first visit, and get an expectation of when we should actually be sending out reviews to the clinicians for conference, when the conference should be occurring, and when ultimately I should be uploading this data to NACC. And not only can we generate these timelines for a prospective subject, we can actually go back and see how well we're doing based off of these expectations for someone who has already gone through the process. In addition, because I talked about conference, we actually do this via a pair of raters to make sure that there is actually consensus on the diagnosis before we fill out the D1. And I built another app that actually, again, connects to our database and will do direct comparisons on responses and identify where there may be issues that need to be resolved and helps facilitate discussion during conference itself. So again, we can see here questions about whether it's MCI or actually dementia for the true diagnosis. I mentioned the dashboard, just to kind of step through this, that I will admit, this does not hold a candle to what the University of Washington has done with Leaf, which is just gorgeous. But this is me doing the best that I can with Shiny via R, and it has worked really, really well for us. So I built a dashboard that actually, again, is connecting to our data in order to get dynamic and up-to-date information in terms of a variety of informative pieces of information that could be used for, say, cohort generation or sample size estimate or some sort of question for a researcher who is filling out a grant application. So we can actually do things, and again, subset by race, which is very, very important for us as a center, since this is our thematic focus, and then get an idea of what kind of diagnoses we have for people who have gone through D1. We're also using it operationally, and even though this isn't actually extracting data, this is kind of letting us know where our cohort stands in terms of people who have been enrolled, who are losing the follow-up, who's asked to be removed or passed away, things of that sort. And we can actually track this longitudinally to see how well we're doing based off of metrics that we want to be keeping based off of our target goal of 50 a year and ideally 100 a year once we get fully funded. In addition, we can also emphasize our recruitment of African Americans because we're really trying to keep it 40%. And yes, I know I said this is generated. We're actually doing a pretty good job of it, believe it or not. I also mentioned the Cody Corps. They like to use this as well because they like to know what events and outreach activities are actually being the most beneficial in bringing people in. And we can do that same sort of subsetting by race as you see here at the bottom and see in particular how well things like our community events are doing and where we want to kind of allocate our resources. And then finally, we also include more dynamic information such as inventories of our biomarker samples that we keep on site. So when we have data use requests, we can actually inform our collaborators what they can expect in terms of things like CSF, DNA, plasma, and the like. So how has this worked? It's actually worked really, really well, I'm glad to say. Working with the University of Illinois, our NCRED GWAS coverage has gone from 35% in January of this year to 80% as of September. And we actually know where our missing subjects are at this point with ID mapping. And we actually have an expectation of 96% of samples sent to NCRED. In addition, our time from participant visit to NACC upload has been reduced by 59% from that same 9-month time window. And we're now at 71 days, which is pretty good compared to the NACC median of 105. We're doing all this while keeping our packets and error rate at 0%. And we're doing this while keeping our enrollment of Black and African Americans at 35 to 40%. And come on. And just to show, again, we all get these monthly reports and there's where we are. We're doing pretty good, I like to say, in the grand scheme of things. So looking to the future, just to tie things up here, again, I mentioned we're really trying to build out a collaborative center. One of the other strengths of REDCap is its survey utilization, especially public surveys. And we've actually built a data use request survey that anyone can access and actually go in so that we can get an idea of what people are looking for within our cohort and what we'll be able to provide and give them a meaningful idea of what they can actually expect from us. And this really kind of helps with our executive committee. And then it has paid off. We actually already have collaborations in sight, and you can kind of see that we have them both within Alabama, within UAB, also at Tuscaloosa at the actual University of Alabama, as well as other ADRCs and institutions like UCSF, Duke, and Washington University. There we go. And then finally, just to touch on this, discussions with other sites have recommended the use of a registry. I like to think of this as widening the funnel so that we have an earlier point of origination for our participants. We can actually contact them for informational purposes at this point, not just research. And this is going to go a long way towards recruitment. So again, our kind of goals as we look towards our P30 submission next year, making the tools work even better, our continued engagement as a NACC and with NACC and NIA as a center, which we're trying to do the best that we can, and just kind of reinforcing that core structure, going from our 2 exploratory cores to just the fully fleshed out P30s. Yeah, acknowledgements, and I know I'm a little long, so any questions?
We have a couple of questions from the virtual participants. Okay.
Yeah, I'll go ahead, log into Zoom and answer in chat the best I can.
Okay.
So.
So That's better. Should we do one question here?
Oh.
Thank you. So so for the inventory, so you have all these different numbers. Who is entering those data?
So we send our samples, we have what's called the span bank within UAB. They actually do all of our processing and storage for us. They send us reports, which we are then able to do direct uploads to our database. And that's what's actually getting queried. So that's how we know who is getting what. We, as a center, don't actually do that data entry step. So
Okay, so UAB has a facility doing that.
correct, yes.
Thank you.
Thank you, Chad. That was incredible on so many different levels. First, for directly addressing the theme of the meeting, which is diversity, equity, and inclusion, and also as a wonderful example how like the data core can help with like data-driven decisions to shape the clinical core of the ADRC. I know that many of you had questions, but Chad's available here throughout the meeting, so please don't let this time limit get into the way of talking with Chad. And he's also a key member of the working group, and he'll be presenting later on what he's done with the all the incredible effort that went into the EDC working group. All right, so it's my pleasure to welcome the next speaker, Dr. Vijay Kolachalama, a close colleague, friend, and a fellow Bostonian. He'll be talking about the evaluation of machine learning readiness in Alzheimer's disease data cohorts.
Thank you for inviting me to give a talk. This is my first in-person ADRC meeting. This is my first in-person ADRC meeting, and last year I gave it on Zoom. So I'm also relatively new to the whole Alzheimer's world. Can you hear me? All right. So as a means of introduction, we are a group of computer scientists, and we've been very fortunate to work with a multidisciplinary team of trainees. Most of them are getting their PhDs in computer science, and some of them are getting their MD degrees in medical school. So we've been very lucky to have these individuals. And as a group, we've been kind of really trying to think about how to sort of really develop machine learning driven or data driven assistive tools that can be helpful in neurology clinics. We have, like I said, trainees from different backgrounds. And as a result, we've been able to sort of really think very deeply about how to sort of develop innovative methods and also sort of really validate them. These are a couple of examples where we were able to sort of really think about how to build interpretable machine learning approaches, as well as sort of really coming up with advanced methodologies that can combine multiple forms of data. But today, I'm actually going to talk about this whole idea of data readiness in the field of machine learning. It's a growing topic. I think everybody's sort of really thinking about this thing. And I think the whole idea is, if you start really thinking about how to build machine learning algorithms, how can we sort of make sure that the data that you're actually collecting is good enough, or is the data ready enough for us to sort of really do machine learning? And I think, just to give a brief background, I think machine learning broadly involves collecting a lot of data, creating models, and building some predictions. And there is a consensus agreement that the quality of the prediction sort of really depends on not just building good models, but also having high quality data. And this number was, my student came up with this number, so don't count me on that. So they said scientists spend about 3/4 of their time in iteratively processing data, which is a lot of time, right? And when we talk about pre-processing data, when we talk about cleaning the data, we want to make sure we validate the data, and we sort of somehow figure out a way to transform the data. So essentially, data readiness is not just a one single step. It's a form of going through many different steps, starting from this so-called raw data, And once you have a raw data, you sort of go through all these steps. Like, for example, you do data quality measurement, you sort of understand, you sort of explain, and you sort of really think about recommending what kinds of data to use to the next step. Sometimes you sort of clean up the data. And in that process, you sort of also do this kind of data pre-processing, and then somehow, and the next step would be to sort of extract all these interesting features from the data. And then finally, you say, okay, we are ready to build some machine learning models. So that's kind of really the consensus. But on the other hand, if you really think about how to evaluate data quality. So again, we did some literature search on this topic, and then we found all these keywords. And whenever somebody's looking at data, they talk about, how to sort of really process the data. So they look at these things called as class overlap, label purity, class parity, feature relevance, and so on and so forth. It's a long list here. So apparently when somebody is spending three-fourths of their time doing data processing, they're sort of really looking at all these different steps. But I think one of the interesting things that we observed is that as we really start thinking about building models, right? So what are we doing? We sort of really first look at the data. We sort of assume that we have to do this kind of data quality step. And then once we do the data quality step, and then we start thinking about building machine learning models. So essentially, most people are sort of really evaluating all these quality metrics more or less independently and assuming that sort of poor quality affects outcomes, which is a very fair assumption. There's nothing wrong with that assumption. But on the other hand, we thought maybe we'll ask the question slightly differently. What if we actually evaluate data quality by defining some sort of an objective, and at the same time looking at the overall model performance? In other words, how can we sort of really evaluate data quality and see if data quality analysis actually is going to affect the actual model performance, and do both those things at the same time, right? So that's kind of really the framework that we've been developing. And in that process, again, I just want to give a quick overview. We sort of really took the Alzheimer's disease neuroimaging initiative data set, and then we started sort of really looking at what's actually out there, right? Of course, there's huge data sets, including imaging and non-imaging, but we sort of really extracted all the Excel files from the ADNI cohort. And then we started to look at what people call as this unstructured data. And then we went through all these different steps to sort of really understand how to sort of really determine outliers, missing values, correlated features, noisy data. And then we sort of really understanding, okay, how to sort of evaluate the quality on these different data sets. And then we came up with an interesting strategy. What we thought was, okay, instead of really using those measures that I have previously shown, what if we actually come up with a machine learning algorithm that can actually evaluate the data quality itself while actually looking at the objective, which is sort of really this classifying the disease. So what we did was we took this master data set, we call it as data set of N by M. It's a matrix, a big matrix of data with multiple different features. C1, C2, and CM are the features that are coming in from these cohorts. And rows are basically the patient data. And then what we did was we randomly started selecting subtables from this large master table. And then we created a lot of different random subtables from that master sheet. So on the right, what you see is this subtable, which is like Ni by Mi matrix. These subtables were randomly extracted from the large data space. And once each subtable was extracted, we actually evaluated data quality on those subtables, and then use those data quality measures as features to actually train a regressor, right? So in that way, what we are trying to do here is we are trying to actually evaluate the data quality metrics and use them as features to train a machine learning classifier that's going to allow us to sort of really evaluate automatically the quality of the data. And again, just to expand more on this, the subtables were created. And we sort of evaluated the quality measures as features based on the subtables that were extracted. And then ultimately, each subtable would actually have a classification accuracy or a performance. And that was also included as the outcome, which was actually trained to sort of really get the weights of those different quality of subtables. So in other words, what we did here was we sort of really took this master table, we sort of extracted randomly multiple different subtables. And for each of those subtables, we evaluated automatically certain quality measures, and then use those quality measures as features to train a regressor that would actually predict the accuracy of that specific subtable. And it's actually an interesting mathematical problem because the question is how many random subtables are you going to select from this master table, right? And it turns out it's actually an NP complete problem, which is very similar to subset sub problem in the mathematical world. And here we did this on the ADNI data set. And then we ran this algorithm by randomly selecting about 500 different subtables from the master table. And this is the data set actually containing all the features from the ADNI data set. So these are the features that we got from ADNI, but these are actually not the features that we used to train the model. The features are actually the quality measures that were computed on these ADNI subtables. In that way, what happened was we did this 4 times. We ran this, and every time we ran, we selected 500 randomly selected subtables. And then we sort of simply asked this question: can you sort of train a model that can predict, these 4 different subtypes, cognitively normal, progressive MCI, stable MCI, and AD. And then the data quality features that were automatically extracted from those subtables were these 5, which is Pearson correlation on those subtables, missing values, Spearman correlation, outliers, and class overlap. And then finally, the outcome was this performance of the classifier on that specific subtable. And then we use this random forest regression to sort of use these features as inputs and then train for the accuracy of the model. And then finally, we saw some interesting results. So what you see here is a table that is summarizing the mean weights that were computed based on the classifier. And then these weights are basically corresponding to all those 5 different features. And then interestingly, it turns out that as you start computing these features from these different runs, we are able to sort of really qualify or at least come up with specific quality values for each of those subtables that would allow us to give us maximum performance on the classifier itself. And it turns out this way we can actually sort of really bring in the objective of sort of classifying the disease as part of this quality assessment process. And in summary, I think what we would like to say here is that data readiness is an important measure to evaluate. And of course, it has many, many different aspects related to data quality. What we essentially did was we came up with a data readiness framework to evaluate AD cohorts, but with an objective to sort of really improve dementia assessment as opposed to sort of really independently evaluating the data quality itself. And there is a limitation, obviously, because if you want to build such frameworks to evaluate data readiness, we need to sort of really think about the objective. Why are we actually doing this? If you just simply do data quality assessment without actually thinking about the downstream outcome, then it sort of really becomes an independent process, and it may not be connected with the outcome. And finally, I think this is sort of really one message that I want to sort of share with you. We come from the machine learning world, and I think there's a lot of work that is being done in terms of data readiness, quality assessments, and all those things. But as we sort of really start pushing further on creating better quality data sets, I think the only limitation is that we are only going to create very nice looking machine learning models. They are probably not going to be true AI-based systems. But I think that's something that I'm happy to discuss with anyone here. Thank you.
Any questions?
This is really interesting. I'll try to understand what is the kind of like assumption here. So you're saying correlation could be a measure to a data quantity.
Correct.
Is the higher means the better?
Feature, yes.
Is a higher a better quantity or lower a better quantity?
So that is up to the objective here. Like, for example, again, the assumption is that if you have high correlation, maybe that is going to somehow help us. But I think we are not making any assumptions on high or low values of the features. We're simply computing all these so-called quality measures, and we are using them as inputs to actually train a classifier. And I think ultimately, if the objective is to sort of really come up with a machine learning model that's going to do high or perform better on a data set, then that should be the objective. So what we're doing is we're computing all these quality measures. Whether they are high or low, we don't really pay attention at that point. We are using them as features to that input to that machine learning model.
So the correlation is as a data set level. When you try to predict people,
right.
whether they're MCI or AD,
Correct.
that's at person level.
Correct.
How you're going to get that data set level information to?
So I don't know how to go back in this. Yeah, this one. So just to make, maybe I should have clarified. So what we are doing here, each is actually a data set.
All right.
So we took a master data set and then we randomly created multiple subsets of data sets itself, right? And we are evaluating quality measures, or we're deriving those features on each of those data sets. And each of that data set, if you run a machine learning algorithm on it, it actually has a performance. So that becomes the output. And the data quality measures of that subtable, which is the data set itself, actually becomes the input features to that training classifier.
Right.
So essentially, we're doing machine learning for machine learning analysis.
I see. Right. Great. Thank you. Yeah. Very
Yeah,
interesting.
thanks.
Moving on, and again, Vijay is available for answering questions online. Please post them. Thank you, Vijay, for that wonderful talk. How cool and clever to use machine learning for machine learning readiness. And again, yes, let's continue this conversation. Vijay is here throughout this fall meeting, and also available, online. Our next speaker today is Dr. Yuriko Katsumata from the UK ADRC. And they're going to be talking about application of item response theory to the study of mixed neuropathologies in the aging brain. Welcome.
Thank you for the introduction, and thank you for giving me this great opportunity. So first, this study is a part of NACC New Investigator Award entitled Pathway-Specific Polygenic Risk Scores on Mixed Neuropathologies. Dr. Further, Dr. Abner, Dr. Nelson are my mentor, and Dr. Van Eldek is the actual PI of this project, and then director of Sanders Brown Center on Aging. I wanted to express my appreciation for them who already support me. Here is our motivation of this study. Our interest is the association between genetics and the neuropathology. But we mainly have two issues. First one is the sample size for genetic analysis of mixed pathology. And the second one is the definition of mixed pathology. The sample size of GWAS for clinical diagnosis AD is getting larger and larger. So one paper published in 2021 had about more than 1 million participants. However, the sample size of the ADSP, Alzheimer's disease sequence project for genome sequence data, and ADGC, Alzheimer's disease genetic consortium genotype data, both are linked to NACC neuropathology data, had a much smaller sample size, which is the for genome sequence data had 1,000 participants, and the ADGC genotype data had 2,000, around 2,000 participants. The next issue is the definition of mixed pathology. So we recently reported that association between non-clinical AD-related synapse and proteopathies, although we showed in this paper, some synapses were associated with other neuropathology than AD pathology, we assume that the mixed pathology may have a different genetic architectures. So this is a list of the neuropathology that we extracted from NACC neuropathology data set. So there are 13 neuropathology that we used. So can you guess how many ways there are in terms of combinations? So, so the answer is 786,432 combinations. That means that if we handled individual combinations, we would have one outcome with 786,000 categories. So of course, it is impossible to do genetic analysis for such outcome. So one of the best solutions to address this issue is to use the dimensionality reduction approach, which create a new outcome variable summarizing mixed pathology. So these are examples of the dimensional reduction approach: principal component analysis, corresponding analysis, linear discriminant analysis, t-SNE, and UMAP. So we successfully applied UMAP for mixed pathology before, and we identified 4 clusters in people with TDP43 pathology. So this method can be used for summarizing mixed pathology. But in this study, we examined a potential of another dimensionality reduction method, which called item response theory. So item response theory is used for scoring a test, for example, mass test. So the item response theory has the assumption that individual mass ability cannot be directly observed. Instead, mass test score can be directly observed. Then the mass test score reflects the mass latent ability. So if there are the 5 problems in the mass test, and the student one correctly answered first and third and fourth problem, and the student 2 correctly answered first and fourth and fifth problems, the total score is the same. But if the item 5 is the most difficult question, student 2 should have more mass ability compared to student 1. What? And the similar way, we assumed mixed pathology condition cannot be directly observed, but individual pathology can be directly observed at autopsy. The case 1 had the pathology 1 and 3, 4 at autopsy, and the case 2 had the pathology 1 and 4 and 5 at autopsy. But if pathology 5 is more severe pathology, case 2 should have more severe than case 1. The property of item response theory is that both item and persons are located on the same latent variable continuum, and the distance between the person and item location here is representing theta I minus beta j is the most important factor. The probability the person I correctly answered the person item j can be expressed using a certain function here. So what does the function look like? The probability becomes S-shape. What? The probability becomes S-shape, which look like a logistic function or cumulative distribution function of normal distribution. So using the logistic function, the S-shape this S shape can be expressed as this formula, which is called Rush model. So in general, so in general, Rush model can be written as using a likelihood function. The Rush model also can be extended from binary to ordered outcome, from unidimensional to multidimensional model, from one parameter to two or more parameter model. In this study, we used the multidimensional partial credit model implemented by the MART-R package to handle this extension. So again, this is a list of the neuropathology from NACC NP data set we used in this study. So there are 1,222 participants who died after 60 years or older and had no missing data. The first, we performed leave one out cross-validation to determine the number of factors, that is, number of dimensions. Based on the sample size adjusted BIC, the three-factor model should be more appropriate. And then, likelihood ratio tests show that the three-factor and four-factor model were equivalent. So first, we ran the three-factor model. The first factor had more contribution from TDP43. And the second factor had a contribution from cerebrovascular diseases. And the third factor had a contribution from AD pathology. The correlation between factor 1 and factor 2 is small, which is the 0.068. And then the correlation between factor 1 and factor 3 is a mild 0.281. And the correlation between factor 2 and factor 3 is negative 0.167. Okay. Yeah. Since the neuropathology was observed at autopsy, the factor score should be related to age at death. Therefore, we made a scatter plot with the x-axis of age at death and the y-axis of each factor score. The factor 1, which is related to TDP43, looks like no association with age at death. And the factor 2 for CVD looks like linearly associated with age at death. And the factor 3, that is interesting, the AD-related risk score looks like there are the two groups. One is the lower group, that there are no association between age and death. And then other group is linearly associated with age and death. Then person who had this mixed pathology pattern is displayed on the red dot. So this person had a high AD pathology. That's why factor 3 is the high score, and the TDP43 is also high score. And then factor 2 related to CVD is a middle score because the cerebrovascular pathology pattern is 31100. Then another example is that this person has less neuropathology, no AD, no TDP, and no HS, and then mild CVD. That's why the factor 1 is a lower score, and then factor 2 is also low, and then factor 3 is a low score. But we realize Lewy body pathology is not reflected into the split plot. Therefore, we additionally run the 4-factor model Then factor 4 is the contribution from Lewy body. As we expected, factor 4 score is very high in this person. In summary, we detected 4 dimensions with TDP-43 rate, CVD, AD, and Lewy body predominant pathologies. Age at death is an important factor to be taken into account, especially in AD and the CVD pathology. So we estimated individual quantitative risk scores with 4 dimensions, which may allow us to do the more powerful genetic analysis compared to binary or category outcome. So we will examine the association between the 4-dimensional risk score and the genetics in future study. Thank you for your attention. I'm happy to take any questions.
Questions?
Sue Lurgans-Rush. This is a very nice presentation. I'm not surprised that Lewy bodies are acting unrelated to anything. That's very interesting to see. I would comment, though, that it does seem that very, very often, even though we know that these pathologies are developing over time in individual people, that somehow knowing that somebody died is so informative about the state of their pathology that it's not uncommon to see no association with age. So I think that's more a feature of how we get to have the autopsy and the condition of the brain. So I wouldn't look at the age association as telling you something interesting.
Yeah.
That's very nice.
Thank you for your comment. Yeah.
So,
We have one question online.
okay, just one from the audience and actually relate to what Sue said. You use the RAS model, which is a one-dimensional IRT, but what have you considered 2-dimensionals? Because you have only, difficulty, but not discrimination. So for example, you may have age could be something that is, easy item, but you could discriminate a lot.
Actually, I didn't show that. Yeah, so I didn't show that. Actually, I ran the 2 or more 3-parameter, 2-parameter model, difficulty and the discrimination parameters. So, but I didn't show this parameter estimate in this presentation.
Thank you, Dr. Katsumata. That was incredible, and explaining to us, difficult concepts such as item response theory to us mere mortals. That was very nicely done. Next speaker is Jacob Fiala. He's going to be talking about the demographic adjustment is not demographic correction, a simulation study. Welcome.
Hello, thank you for giving me the opportunity to speak. So today I'm going to talk about 2 sources of demographic differences in cognitive test scores. Move this up a little bit. There we go. Let me briefly remind us of the concept of test norming or demographic adjustment. So for example, adjusting cognitive test scores for a factor like education reflects the assumption that we can't expect the same level of performance between, for example, low education group versus high education group. And moreover, it has the assumption that the difference between those groups is strictly due to bias or error. And if we can get the education effect out, then the test is then fair for both high education and low education individuals. Unfortunately, demographic adjustment can remove 2 kinds of variance from cognitive test scores. Differences caused by measurement bias, but also differences that reflect different levels of exposure to risk factors. So measurement bias refers to demographic group differences that would not be present if the test was not biased. It's by definition error. In contrast, demographic group differences can be caused by relative risk differences. A relative risk is a statistic that tells you how many times more likely one group is of experiencing an issue compared to another group. So for example, a relative risk of 2 means one group is twice as likely to experience the issue compared to the other group. So relative risk factors, the differences arise because of differential exposure to risk factors like on the slide there. So this can lead to differences in average test scores as well. As you can see there below, a large relative risk difference when you use random sampling would then produce that the group with higher risk would then have higher prevalence, which would mean more low scores in the group, which would drag the group mean down. This is a problem for demographic adjustment because it's likely that the demographic factors that we are adjusting out are due to both measurement bias and relative risk imbalance. But when we adjust for, oops, it is frozen. Oh, there we go. But when we adjust for a variable like education, we are not distinguishing between those two sources. And so we are removing both types of variance. And as the visual metaphor of the slide, it shows we are throwing the baby true differences in risk out with the bathwater, which is bias. So recognizing this problem, I was left with 3 questions. First, in what conditions does demographic adjustment improve diagnostic accuracy, and under what conditions is it counterproductive? Second, is adjusting for risk factors themselves a viable alternative to using the demographic groups? And finally, what test score adjustment methodology works best under all conditions? So I will now describe my methodology, if it will let me. Okay. So to answer these questions, a simulation study was needed. Simulation studies allow us to investigate things that would be impossible or unfeasible to study with real life data. So they allow us to vary parameter values across iterations to make sure they're widely applicable, the findings. And in this study, it provided error-free knowledge about the true impairment status of each participant, the true prevalence of cognitive impairment in the samples, the true exact amount of measurement bias, and each participant's individual risk profile, which you can think of as a composite of risk factors, kind of like the Framingham Heart Risk Score, but for cognitive impairment. There were 12 parameters in the simulation, but we only have time to discuss the 5 key parameters. The prevalence and average level of risk in the groups, which are meant to represent different levels of exposure to risk factors, were determined by 2 parameters. First, the high education group, their risk level was set to a predefined level, and then a relative risk parameter defined the low educations group as a multiple of the high educations group. And then the 3 remaining key parameters are all different facets of measurement bias. Measurement bias can affect test scores in a variety of ways. For example, measurement bias could cause one group to miss several questions unfairly. That would be represented by the mean measurement bias parameter, which deducted a certain amount of points from one group's scores. Measurement bias could also impact variance, either inflating it or reducing it. And that was represented by that variance measurement bias parameter. And also measurement bias could cause a test to be a less valid indicator of impairment for one group than another group. And that was represented by the validity measurement bias parameter, which reduced the correlation between test score and true impairment status for the low education group. All right. Before we discuss results, let's overview the procedure from a bird's eye view. It actually, okay. It actually took 414,720 iterations, and the supercomputer at UF 2 weeks to run through this. So this procedure was repeated many times, but first step was generating the normative sample, including a true score that's completely error-free, and then the raw score, which has whatever level of bias that iteration has embedded into it, and the risk profile score. The second step was still, okay. The second step was adjusting the raw scores based on different variables, which you can see on the slide. And then the final step, calculating a lot of statistics to evaluate the effectiveness of each set of scores. In this presentation, we'll focus on score means and also the accuracy, sensitivity, and specificity of each set of scores determined by an ideal cutoff from an ROC analysis. Okay, this brings us to results. And first, we'll look at the results pertaining to question 1. When is demographic adjustment beneficial and when is it counterproductive? Before we look at which conditions specifically are best and worst suited, let's look at the overall results across all those iterations. I'll show a few tables like this, so it's worth pausing to kind of explain the layout. That first row there is the true scores accuracy. So the other rows are how they compare to the true score. So for example, the raw score has surprisingly good full sample accuracy, 3% lower than the true score. But notice there is a significant differential in accuracy between the two groups, which was not present in the true score. We see similarly down below with sensitivity and specificity that the raw score achieved its accuracy by being more sensitive to the low education group and less specific. And conversely, in the high education group, it was less sensitive and more specific, meaning that in the low education group, impaired people are less likely to be detected by the tests. Demographic adjustment did restore an equal level of specificity, but it came at a sensitivity cost for the low education group. So now we'll look at the means of test scores within the groups. All scores in this table are on a Z-score metric, so they would be comparable. So it looks like demographic adjustment fixed all the bias, right? Actually, no. So looking at the true score there actually should be a demographic difference in mean scores remaining. It's not due to measurement bias. It's due to different levels of prevalence in cognitive impairment in the samples. So this can be seen in the bottom table, which stratifies by the impairment status. And you can see with the true score, there is no education difference to be found. The normal cognition has the same score and impaired cognition has the same score. But when we look at the demographic adjustment, we see that the low education scores have been inflated to offset the higher number of impaired people in the group. So what conditions is demographic adjustment more suited for or less suited for? To test that, we did a linear regression, and the outcome variable was the difference in accuracy after adjustment from the raw biased score, the unadjusted score. Predictors included the key parameters, and I can only show an abbreviated form of the results due to time constraints. But we found that Iterations with high levels of measurement bias had more improvement in diagnostic accuracy compared to the raw scores. But iterations with high levels of relative risk difference were actually reducing the diagnostic accuracy, even compared to the raw biased score. So the takeaway is when group differences in the cognitive test score are due to measurement bias, demographic adjustment is helpful. And when they are really primarily due to relative risk differences, it can actually be detrimental and hurt the accuracy of the test. In practice, that's definitely a terrible outcome to hurt the accuracy of the test. So looking at real life situations when presumably both types are present to some degree, to see how it would perform under that, we have to look at the interaction between the relative risk variable and the measurement bias variables. And we have a graph here for the mean measurement bias interacting with the relative risk. And the first thing to note there is that top red line is when the risk is equal between the groups. And you can see clearly as measurement bias increases, so does the improvement in diagnostic accuracy from adjusting scores. But when we get to the lower levels, as we go lower and lower, meaning that as there's a bigger relative risk difference between the groups, the improvement becomes more and more negligible, with the last line remaining below zero the entire time, meaning counterproductive in all cases. To iron that home, this is the stratified means again. This is in a situation with equal risk, and you see demographic adjustment essentially completely restored this correct means. But when we have very high relative risk difference, we see the same pattern as before, inflated scores in the low education group to offset the increased number of impaired people. It brings us to question 2. Can adjusting for risk factors instead be a viable alternative? Unfortunately, the answer is unambiguously no. It hurt accuracy, sensitivity, and specificity for both demographic groups. And the degree to which it hurt those was correlated inversely to how the signal to noise ratio in the risk profile. So as it was less noisy, it did more damage. Okay. So those keeping track know that there's a third question, but we actually do not have time for it. And it's ongoing. So I will address that in the next section, which is implications in future research. To recap, we have we found that the demographic adjustment was only beneficial when the degree of measurement bias was sufficiently high and the relative risk was sufficiently low. This is an enormous implication considering the ubiquitous practice of age norming. There is a well-established huge relative risk difference between age groups. And it also underscores that future research is desperately needed because we do not know to what degree prevalence differences were present inside the samples that produced our widely used norms. Regarding the second question, we found that risk adjustment was inherently counterproductive. I know this goes against kind of common wisdom, but it does make sense. In the same way that demographic adjustment equalizes scores between demographic groups, risk adjustment is equalizing scores between those who are most likely impaired and those who are most likely not impaired. So trying to use the test to differentiate those groups after you've removed those differences is counterproductive. I don't think I have time for my final method here, but I will say that the opposite of risk adjustment, where we combine risk with the score, like a risk-informed test score, has in the current manifestation, in the simulation, I mean, like its current form, using information that would be available in real life situations, improve diagnostic accuracy even over and above the true error-free score. because new information is available through the risk factor. And it worked pretty well at better than the other methods at reducing demographic bias. So in future research, I will use UDS to construct a risk profile in real life. I will use it to further refine my methodology and cross-validate it using real data, comparing that to traditional methods. Thank you.
Great job, Jacob. That very exciting. One thing that you mentioned, and I think can kind of go a long way, is you mentioned you started with 12 at the very beginning. This gets into something that we often run into with covariate adjustment, which is we toss everything into the model and then hope for the best. And what we often wind up with is that a lot of these variables wind up competing against each other. You have variance inflation. And one of the ways around this would be something like an instrumental variable approach. And I can see this being a really good avenue to address that, to see if that would also be an option to really kind of tease some of this out, since you can bake that in to the simulations. Have you had any thoughts about trying to enable that to see if that actually improves this kind of performance?
Yeah, I'm sorry, through speeding through this like months of research into 13 minutes, the 12 parameters were kind of varying conditions, like the sample sizes and all these different things that could arise in testing situations. They weren't variables within the norming equations. The demographic adjustment was simply the group. And then risk adjustment was that risk profile score I mentioned, that is like a Framingham-Hart risk score for cognitive impairment. And there's so many parameters and ins and outs that I didn't have time to go into, but the norming itself followed the conventions in the field and was relatively simple.
Okay, great. Really look forward to seeing where this goes.
Thank you. Got one over there.
Beautiful job.
Thank you.
So have you considered the possibility of circularity in the data, for example, that the risk factors that you are analyzing here are also highly correlated with multiple of other instances that you are analyzing? So whenever you correct by the complex risk factor, you are, imagine these are genes, for example. If these are genes, the genes you are correcting for multiple, for example, a PGRS, maybe containing not only ApoE4, but it's also containing intelligence-related genes. So whenever you are correcting for this, you are also overcorrecting by the other factor. For example, the socioeconomic factor are impacted by this because, okay, you are getting people who is related to the other, to lower statuses. So consider circularity, and maybe you have to dissect.
Yes,
those things.
absolutely. Of course. I will answer that over the break. It gets into the 12 parameters.
Sounds good. Thank you, guys. I will start. Thank you for inviting me to present in our work at the NACC data core workshops. Especially, I'm the first virtual speakers. Hello, everyone. My name is Dama. I'm an assistant professor at Wake Forest University School of Medicine, ADRC centers. So today, I'll be presenting our recent work on the estimation to the time to conversion for Alzheimer's disease using a neuroimaging genomic approach through a multimodal deep survival analysis. So here's some background about the survival analysis. As described in the titles, in this study, we aim to predict the time to future dementia conversion for those patients at their earlier stage of the disease progression, using multi-type predictors as biomarkers. And we also aim to study the influence or the importance of each data modalities using these predictive tasks. So those modalities including the neuroimaging data in terms of MRI the genomic data in terms of the SNPs, the single nucleotide polymorphisms, and also the cerebral spinal fluid biomarkers in terms of the amyloid and tau concentrations in the CSF, and as well as other variable cognitive tests. So we approach this problem through the survival analysis, and we already heard in the morning that it's a time to event analysis. And this is a set of the statistical methods to estimate the probabilities of either an individual or a population who will experience an event of interest at the specific future times. And in our case, the event of interest is a dementia onset, and we model these probabilities as a function of time. So the figure on the right shows an example of such survival curve, this representing the age-related risks for developing Alzheimer's disease with different type of ApoE allele at the populational level. So for example, here, the higher survival rate represent a higher chance of staying healthy and cognitively normal. And when the survival rate drops, like shown in the figures, that means the group are more susceptible to developing dementias. Specifically, if we look at the purple survival curve at the top of the figure, which indicate a significantly lower likelihood of developing Alzheimer's disease for those ApoE2 homozygous, which means those people are with 2 copies of the ApoE alleles will have a much lower risk compared to the populations in other groups. So in the case of our study, we aim to construct the survival curves not on the population level, but on the individual levels. We achieve this through a personalized prediction models buildings. And compared to the traditional regression-based methods, which can also estimate the event onset time, the survival analysis can account for individuals who has not progressed to the dementias during the follow-up visits. So this allows us to utilize all the available data, although we know there is still limitations about such approach. So in survival analysis, this kind of the missing data or about the event onset for some subjects during the study period are called the censorship. So for example, as shown in this figure on the right, the patient who were diagnosed as dementias before the study started is called the left censored. And the right censored include those subjects who are cognitively normal subjects who might develop dementias beyond the study period. So we haven't measured them during the study, between the study start and end point. All those healthy subjects who are still in the middle of their following up, so they haven't finished their follow-up time point measurements. So we developed and validated our models using the longitudinal follow-up data, which were collected from the ADNI-1 datasets as a proof of concept studies. And the table on the left shows the study participants. We divided them into 2 categories based on whether or not they receive a clinical diagnosis of dementias of Alzheimer's type during the study period. The non-progression categories include the subject who did not receive a clinical diagnosis of AD throughout the entire study period. On the other hand, the progressive categories, or we call it uncensored data, who includes those subjects who received the Alzheimer's disease after their initial visit and during the study windows. So to evaluate our models, we split the data into training, validation, and testing sets. And just to notice specifically that we stratify the data splittions, which means for each partition of the data set splitting, it contains the same sample ratios across different subgroups, which was defined in the tables on the left. And also want to note that only the baseline data were used for the training because we will only use the follow-up data as the defining the ground truth labels. And here's the outcome measurements that were used to build the prediction models. The ground truth labels for each subject in the studies are split into 2 parts. First is the event indicator, which is either 0 or 1. Reflecting whether a subject was progressive means 1 or non-progressive means 0s. And also the second part is the duration. So for progressive subjects, this is a time between the first visit and the confirmed dementia diagnosis in the follow-ups visits. And for the non-progressive subjects, the duration simply means the time between the first and their last visits. So now let's look at the input data types. So first, there are 2 data types which are high dimensional in the ADNI data. First is the neuroimaging data, and the second is the genomic data. So they were first pre-processed and the features selected. And for the MRI data, which is the neural image data, we using the free surface segmented volumes, and a total of 91 volumes were parcellated, and we control that for the total integrated volumes. And for the genomic data, about 500,000 of the SNPs data, the single nucleotide polymorphism data, were derived after the quality controls using the P-link pipelines. So both features were then ranked and selected based on the statistical test for both the continuous or categorical variables according to the types. So MRI is a continuous and the SNPs is a categorical variables. And then we repeated this feature selection process and by resampling the splittings by 10 times through the sub-bagging process to make a more robust final 21 selection of features vectors, which are derived from both data types. We then concatenated them together. And here is a final set of the multi-type feature variables. A total of 21 feature vectors were selected from both from each of the 3 feature types. This includes the neuroimaging, the genomics, and as well as the 20 features from the as a composition of the demographics, the cognitive test, and the CSF. So here, after we define the outcome measurement and as well as the input measurements, here is the detail about the deep survival model design architectures. So conventional survival analysis could be achieved through a special regression models called cognitive regression, or COG regression, or the COG proportional hazard regression. So basically, the model is trying to model the probabilities of a hazard event at a specific time point in the future, hence the name hazard model. And it is calculated as a function of a linear combination from multiple input variables, as indicated as the g of x here, shown in golden. The x here is the vectors of multi-type features in our study. And the final survival estimations is derived from the cumulated hazard functions. In order to capture the non-linearity or non-linear relationship between different feature types or within the feature types, we use a deep learning-based multi-layer perceptron or MLP architectures to derive the same hazard functions. We substitute that in the place of the original linear functions used in the traditional Cox regression models. So the deep survival models was then trained and minimized, trained by minimizing the loss functions as shown in the bottom. This is event indicator D, which reflects the progression versus non-progression, so 0 or 1. And also there is a negative log likelihood of the hazard functions that attach to the loss functions. So to compare and determine the effect of each individual features, and also to look at the combination of their effects towards the survival analysis, we conducted a set of different experiments using different combination of feature types as input predictors. So here list 7 of them. So basically it's either single feature types or combination of 2 feature types, as well as all feature types. And then we have to evaluate the model performance. So here is a first measurement or the first evaluation metrics to measure the overall performance of the models. And it's called the integrated Breyer scores. This is to measure the average square distance between the observed and the predicted survival probabilities. Since it's a distance, so the smaller the IBS scores represent a better predictive accuracy. And here shows the result of the IBS. So basically, I can summarize the result into like 3 observations. First, the dementia, the demographic cognitive test and CSF, the DTC scores, which is shown in green, works the best among the single modality feature sets, which makes sense as the clinical diagnosis of dementia onset. It is largely determined by the cognitive test. We also observed that by combining the MRI and the genetic features, so for example, shown in the red here, it improves the performance of each individual single feature types as shown in the blue and the yellow. This indicates a potential genotype-phenotype interactions through this kind of a multi-type, multi-model feature fusions. And finally, by adding MRI to the GTC feature types, it slightly improved the prediction performance, although actually it's not statistically significant. However, by further adding the genomic information, it did not show improvement, but actually reduced the performance instead. The second evaluation matrix we used is the concordance index or C index. It's basically taking a pair of individuals, derive the order of their survival time, and then compare the true order of these two pairs to the ones that derive from the predictive survival functions. So for the C index, the bigger the better, because it means that the same orders has been achieved through the predictions, and that's a better concordance, hence the better predictions. And here shows the result, and we can summarize that in pretty much the same way as the IVS result shows. So the DTC give the best result among the 3 individual feature sets. Combining MRI and genetics improves individuals, and adding MRIs give a slightly improved in performance, but not the genomics. To make it down to the individual predictive levels, we want to evaluate whether the differences between the predicted and the true dementia conversion times is clinically relevant. So here, for example, the blue dots represent the predicted dementia onset time, which is defined as the time point where there is a 0.5% or 0.5 survival probabilities. And the top rows here shows the non-progressive subjects. So you can see that the green vertical lines represent the right censoring time because it hasn't progressed throughout the study time. And the low rows shows the progressive subjects. For those subjects, the red vertical lines represent the true time when the subject was diagnosed as the Alzheimer's of dementia of Alzheimer's type in the future. So with that information, we could then derive the difference by subtracting the true survival time from the predicted survival time. So this is the outcome of that differential calculations. So for the prediction time, it is the subject's survival probabilities reach 50% as a reminder. So if we look at the results here, and if you can see the 0 means that there is a basically perfect prediction of the future dementia onset time.
Sorry to interrupt. There's a little time check. So if you could wrap this up in the next 10 seconds or so.
Sounds good. Yeah, no problem. So basically this result shows that so there is a large percent of the cases where we can have a best prediction of the survival times. What is more interesting is that I will summarize it here, is that if we separate the cohort into the normal cognitions, as shown on the left, and mild cognitive impairment subjects, the performance is different. So basically, if we look at the genetic samples, it can get better predictions for those normal cognitive subjects, in which case they haven't developed the dementias or diagnosis yet. However, if you're using MRI features, it gives all the DTC features. It cannot detect the features, which makes sense because the syndrome has not derived. And comparatively, so when the subjects are in the mild cognitive impairment stage, the performance is for the MRI and DTC is better. However, the genetics is falling back. So combining them together, it works for the MCI, but not working for the NC, meaning there is a mixed performance for the improvement when adding the genomic informations. So yeah, I will skip the feature importance analysis part due to the time constraints, basically showing that our result is matching with the known knowledge, but also giving more novel knowledge about the genomic-based features. And when combining 3 together, we can see the relative importance of different feature types. So in conclusion, we show that different data types are better at predicting Alzheimer's disease onset time for subjects at different disease stages. For example, genomics better for the progressive NC, and neuroimaging and cognitive tests is better for the progressive MCI. And we can, using the normal AD-related genomic factors to discover through the model cognitive feature importance analysis. And the future work will involve the improvement of the feature extraction method and further validate that through the ADRC and NACC cohorts. So just to mention that.
Thank you. Yeah, thank you, Dr. Ma.
Sorry,
yes, incredible talk. And we unfortunately don't have any time for questions at this point. So please post your questions online. And we're going to go ahead with Dr. Meredith Zotas from South Texas ADRC. She is a clinical research informatics expert and will be talking about measuring alignment between ADRC, UDS data elements, FDA and EHR data standards. And just so that we don't run late from now on, after 10 minutes or so, I'll give you guys a 10 check to wrap up and do these questions. And please, again, this is just the start of our conversations. There's hopefully lots of opportunities to carry on this conversation. Thank you.
Thank you.
Awesome, thanks. So in clinical and translational science, there's been a major roadblock that's been identified, which is operational efficiency in clinical trials. But, you know, that also corresponds to a lot of what we do in observational data collection as well. And this particular project is an intervention to look at both the operational efficiency of data collection at clinical sites, as well as an intervention to take a swing against the largest source of error in data collection, which is medical record abstraction. And in doing it, it's really important that we take a very deliberate approach, because not all of our data are abstracted out of medical records. Not all of us recruit patients in the same way. Not all of our patients have medical record data at our site because they weren't a patient until they came and saw us for their initial evaluation. And so what I'm presenting is a mapping of data between two different data standards of our UDS forms, so that we can take an initial look at how much of the data could we get out of electronic health records at places where that is the source of the information, using the current standards, which are the Health Level 7 Fast Healthcare Interoperability Resources Standards, which is, it's like a spigot on the back end of the electronic health record, where we can query it and get the data out in a standard form. Before the FHIR standards from Health Level 7, that was not really possible at scale, because every institution was sort of a one-off, going to the EHR to get the data. So the first side of the mapping is, what can we get from electronic health records, given that there's data there for our patients? And then the second look is in a totally different direction. It's looking forward to potentially using this data for things like post-market safety surveillance, and which are, for example, if the FDA wanted to look at the safety surveillance of any recently approved drugs. And so to do that, it would really be helpful if the data that the FDA could get from an initiative like this could be provided to them in the standards that they're used to seeing, that they're used to getting in their data viewing tools, and that they're used to working with for secondary analysis and looking at the data. And so what we did is we took the UDS forms, we did UDS 3 and then also the UDS 4 forms that were available. And we had two independent people map them to the health record FHIR standards. And then two different independent people, oh, awesome, thank you. Two different independent people map them. We can just go ahead and, how do I advance them like this? Okay, awesome. Two different people map them over to the CDISC standards, or the Clinical Data Interchange Standards Consortium standards. That's not the talk I was planning on giving. Here, could you guys find them? So, okay, so two people mapped them to the EHR standards, two people matched them to the FDA standards. And then we looked at differences between those, given that we didn't ourselves develop, not a lot anyway, of either one of these standards. We had the authorities for each of the individual standards adjudicate where we disagreed between our two independent mappers. And so the Clinical Data Interchange Standards Consortium folks adjudicated our CDISC mapping, and Miriam Garza, who is one of the co-chairs of the Health Level 7 Clinical Interoperability Council, who's published actually the last two systematic reviews on EHR data acquisition, as well as some FHIR mapping work. And so basically, they adjudicated the results, and the information that we have on the table in the slides, which if we can't see, it's okay, you'll be able to refer to it. The data that we have on the table is basically the adjudicated percent that mapped to each standard. And so what we found is that the CDISC standard going to the FDA is fairly generic. It was constructed as findings, events, and interventions with some subject characteristics that's expandable through an auxiliary data set, and then some other supplemental qualifiers for any of the findings, events, or interventions data sets that's also extendable through the standard. Awesome. And so what we found, the CDISC are the last 4 columns, and then the FIRE are the 2 preceding columns for them. And so what we found is that the FIRE data mapping is significantly a lot lower than the CDISC, because as I mentioned, the CDISC standard is pretty flexible. It's used to putting a lot of stuff in it. The EHR data, from the data that we collect, not as much of it would be gotten from the EHR, as most of you are probably already familiar with. Importantly, in these mapping results, we did count questionnaires as mapping to the electronic health record. And there's a big caveat on that, because whether or not they map to the health record, it doesn't really matter if the health record wasn't used as the source of those information. So the fact that we could put them there, if that workflow works best for a site, and we could get them back out through the standard spigot, is great. But the caveat is that might not be the best workflow for your site. So in looking back at the mapping results, there's a lot of things to consider in there. The first is that when we look at the percentages that map, for example, to FIRE, those look like, from a time savings perspective, they could be helpful, because when we've gone out and talked to clinical investigational sites, they say, good heavens, if you could just get me meds and labs, I'd be really happy. So, these percentages that we're seeing from FIRE, worth a look maybe. And the percentages from CDISC, encouraging, because we may be able to give the data to others who might use it for public health in the standard format that they're used to. And so with moving forward and looking at this, the important thing to think about from these mapping results is they look like, yes, it's worth another look. I think for us, a good question is, what should the next step be? One reason why I like working with groups like this is that they tend to be generally more deliberate in moving forward with things and making sure that it's going to be a good next step. So I think looking at the mapping at multiple sites would be a good next step if anybody's willing to do that. We have put software in the Epic App Orchard for anybody that's on Epic that will allow you to query your FIRE server and look at what the mapping is. So I think that would be a great next step. The other thing that I want to mention is that in doing this and moving forward, a data quality assessment is another next step that needs to be done before. So I don't want anybody to get too excited about this before we talk about actually going out and measuring the sensitivity and the specificity of the data that we would collect through this. And given the multi-sources that are used and the different data collection scenarios at our sites, that will be a very interesting research design discussion. So I presented this today. We'll be happy to share any of the mapping templates if anybody's interested in those. Would love to hear from anybody who's interested in doing site-specific mapping at your site. We generally see a 10% variability in the mapping between sites, but haven't done Alzheimer's at different sites before. And would love to talk to folks. So with that, I'll stop and take questions. There's no need going back to the slides.
That's great, Meredith. Like, you say all this, and the first thing that makes my blood run cold is the mapping that I've had to do from ICD-9 to ICD-10. So I sympathize completely. The thing that worries me the most from what I saw was with those percentages, one of the lowest mappings you had with FIRE was CLD, which is the cultural language and diversity module. And I'm just kind of wondering, what kind of implications do you think this will have with sites that have a high prevalence of patients that are like Spanish speaking and things like that?
Yeah, sure. So that could be us in South Texas. One of the things that we've seen is that people have implemented the EHR socio determinations of health very differently. Some of the EHRs have standard things like the Epic wheel for social determinants, others not so much, and other sites go at it on their own. So it depends on how well that's collected at the site as to whether the EHR is even a reasonable source of that information. What we found when we mapped just race and ethnicity is that, and I've done this at 3 different healthcare institutions, like in different states, and there's about a 30% discrepancy rate, just race and ethnicity. So chances are it's gonna be that bad for those type things, even if it were in the EHR.
Kind
Not a
of
great answer.
reassuring, but
Yeah, not a great answer, but you were thinking the right direction. Thanks, guys.
Thank you, Meredith, for that wonderful talk and for doing this without without any slides and helping through this mini technology crisis that we have had with the hybrid session moving from online to in person. Next speaker today is Dr. Yu Ma, a study on the sample representativeness of the Wisconsin ADRC clinical core participants of the Wisconsin state population. Welcome.
Thank you. Hello, everyone. I'm happy to present our study. Our Wisconsin ADRC participants has been included in many AD studies. Understanding how a participant sample is compared to the population will be informative. Planning, data analysis strategies, and evaluation how generalizable the findings based on the sample to the population. For these considerations, we compared our standard participants against the state population, Wisconsin state population on demographics and vascular risk factors. So our clinical core sample included all the ADRC participants who were active as of November 30, 2021, which was our data freeze cutoff date. And the sample included totally 678 participants, with 296 between age 45 and 64, and 382 between at age 65 and older. We conducted analysis, the comparison for the 2 age groups separately. And for longitudinal variables that included repeated measurements, such as age and vascular risk factors, we used the most recent visit data. And for population statistics, we obtained the statistics from several national and state data sources, data tools. For the age, the population studies on age, sex, race, and ethnicity, we calculated the percentages based on the counts provided by the Wisconsin Interactive Statistics on Health wish query maintained by the Wisconsin Department of Health Services. And the query population module presents population counts and estimates for Wisconsin. And the data are statewide, by region, by county, and also broken down by different demographic groups. The education is based on the Behavioral Risk Factor Surveillance System, BRFSS survey data. And BRFSS include national telephone surveys about US residents on health-related behaviors, chronic health conditions, and use of preventive services. And the surveys collect data in all 50 states, District of Columbia, and 3 US territories, and complete over 400,000 adult interviews annually. The system provides several data analytic tools that can be used to present various graphical presentations or table summaries of the health-related population statistics estimated based on the survey samples. It can also be used to present longitudinal trends over years or comparison of across geographic areas. So the rural-urban distribution was obtained from the Rural Health Information Hub. And this data hub provides many data visualization tools, including interactive charts, graphs, and maps on a variety of topics related to rural health status, healthcare, and disparities. And population studies on vascular risk factors were estimated based on the survey of the Health of Wisconsin show data. Show includes household-based examinations, health surveys on representative samples of Wisconsin residents. The topics include health conditions, health-related behaviors, healthcare, social economic determinants. And 4 waves have been conducted since 2008, including over 6,000 participants. The surveys included self-report data, objective measures, and biological samples such as blood, urine, stools, and DNAs. So we applied several statistical tests to evaluate the difference between the sample versus the population. So for continuous variables, we applied the one sample t-test because population variance is known. If the population variance is not known, we could use the one sample t-test. Binary variables were tested using the exact binomial test, and the categorical variables with more than 2 categories were tested using the exact multinomial test with the Monte Carlo approach. So in the exact test, the exact probability for each possible outcome was calculated based on the sample size in total and each of the categories. And the p-value for the test was calculated by summing up all the exact probability of all the possible outcomes that are less likely than or equal likely to the observed outcome. So as the number of categories increase or the total sample size increase, the exact probability test can become computationally very intensive. And at this situation, the Monte Carlo approach can be used to overcounter the computing difficulty. We applied the false discovery rate correction method for adjust the inflated type random error rate associated with multiple testing. So next, I will present the results. Here shows a comparison of the age distribution of our center participants against the state population of 45 years and older. So each data bar represents a percentage for an age group by 5-year increment, with the blue bars represent the state populations and red bars represent our center participants. We can see for each group younger than 60, our center participants have a lower percentage than the state population. However, for all the age groups older than 60, our participants have a greater percentage. So overall, our center participants are older than the state population. Here shows a comparison of some of the demographic variables, gender, race, and ethnicity. And we conducted analysis and represented results for the 2 age groups, younger versus older, separately. And within each group, there are 2 columns with the left column state population represents a percentage for the state population, and the right column represents the center participants. And we can see for women, for the gender, our center participants have a higher percentage of women than the state population in both age groups. And our center participants also have a greater percentage in the underrepresented minority groups than the state population. More specifically, we have much higher Black and American Indians, but lower percentage of white and Asian. Our center also have less percentage of Hispanics than the state population, although the test did not reach significance. Here shows a comparison in education and rural residents. And we can see, compared to the state population, our center participants are much better educated for both age groups. If we look at the college graduation rates, we can see our center participants have close to 70% who have college degrees. However, in the state population, only 29% of the younger group and 22% of the older group have college degrees. So we also have lower percentage in the city population in rural residents. And if we look at the distribution for the center participants only, we can see the 2 age groups are actually very similar in the distribution for education and rural percentage for our center participants. However, for the state population, we can see the older group are actually less educated and have a greater percentage of rural residents. And because of age difference in population, the sample versus population difference is actually greater in the older group than the younger group. Okay, sure. So this just briefly shows the we can see our center participants have lower hypertension, diabetes percentage, lower blood pressure, and lower BMI, and also similar in a younger group have the total cholesterol, but the older group, our center is greater. So in summary, we can see our center participants compared to the Wisconsin population 45 years and older. We are older, we have more women, greater percentage of Black American Indians, better educated, have less percentage living in rural areas, and have a better vascular risk profiles. So I think this is an important slide. So our center participants, because our research center, the goal is now target for epidemiology studies, our recruitment is now designed to be representative of the population. As an AD research center, we actually target on recruiting older adults with AD and MCI or cognitive impairment with increased AD risks. So as a result, our sample is now suitable for epidemiology studies such as estimation of prevalence rate of AD. Second, we make a great effort to recruit Black American Indians into our center. So as a result, a quarter of our participants are underrepresented minorities. And this oversampling is important for studies that address racial disparities or studies that specifically focus on underrepresented minorities because it helps with alleviate the lack of statistical power associated with small and unbalanced sample sizes that are commonly encountered for these studies. And it's also help increase precision effect size estimates. And in the future, we would like to target recruit more people who are less educated or living in rural areas, because these populations, they have less access to quality healthcare and are more vulnerable at greater risks. So making sure they are included and well represented in our research would allow our research conclusions to be generalizable to these subpopulations, which would eventually help support better policymaking to meet their needs. All right, so I would like to acknowledge collaborators and staff, and most important, our wonderful research participants. Thank you so much for listening.
Do we have any questions? Okay. Thank you. Thank you.
Hello, my name is Dr. Kevin Bieniak, and thank you for attending my talk entitled 3D Brain Modeling and the Prospect of Virtual Reality Neuropathology course. Today I'm going to be talking about 3D brain modeling of the postmortem human brain and where I believe it has interesting applications for neuropathology core practice, research, and education. This is a case we'll be using as an example of 3D modeling. She was a 5 foot 1 inch, 124 pound right-handed Hispanic female with 12 years of formal education who began having onset of disorientation and forgetfulness at the age of 52, at which point she was dismissed from her job. MRI showed bilateral medial temporal lobe atrophy with T2 lesions in the deep frontal, parietal, and periventricular regions. On examination at the age of 54, she demonstrated anxiety, trouble with instrumental activities of daily living, incontinence, hallucinations, resting tremor, and aphasia. She performed poorly, scoring only 3 points on the mini mental state exam, and had FDG PET left temporal parietal hypometabolism compared to frontal. Athena CSF AD mark panel showed elevated A beta 42, total tau, and phospho-tau 181. And this individual died at the age of 55. She had no known family history of neurodegenerative disorder. Here I'm showing our laboratory setup that we use for 3D imaging. In the center we have our Einscan H scanner, which we use for image acquisition. A fairly standard photography, videography setup with a backdrop, LED lighting, and a motorized turntable where we place the brain for imaging. This setup is connected to a workstation that's using a NVIDIA Quadro RTX graphics card, as well as an uninterrupted power supply. And all of the images are stored on a 2 terabyte solid state external hard drive. The files generated from our imaging system are DAT files and then BRAPPRJ files as well. These are Einscan specific file formats. The output size of these files is fairly large. It ranges anywhere from 4 to 12 gigabytes based on the number of scans that we're doing and then aligning into our final image, as well as the resolution settings and the number of unique points that we capture on our object here, the brain that we're imaging. Typically, we're capturing about 20 to 40,000 unique points on each individual scan, and we take several of these that we align into our final brain image. And the ultimate files that result from these are a number of different 3D file formats, including STL, PLY, OBJ, and 3MF files, and these range anywhere from about 1,000 to 12,000 kilobytes. So first I'd like to start with how I think 3D modeling has important implications in neuropathology core practice. Prior to cutting, we scan the intact fixed left hemibrain and then remove the infratentorial tissues, including the brainstem and cerebellum, and lastly, extrude the medial face of the brain down to a flat surface. The extruded model is what we use to generate our brain tissue matrix, which is essentially a box that we use for cutting the brain. And this has slats that are evenly spaced at 5 millimeters, which fits right into our tissue cassettes. And we can label these with the case number and a unique number for each of the different slabs that we'll generate in cutting. We print our brain tissue matrices on a Raise 3D Pro 2 printer. This is a 3D printer with a fairly high build resolution at 10 microns and a large build volume. It also is equipped with dual extruders, which you can use to print with different filament types or different filament colors. Here we're using a single filament polylactic acid being printed in red. And these tissue matrices can take quite a while to be built. Here, this is a timeless video over 36 hours. The upper left-hand image is showing a completed print, which is labeled with the case ID number BI22035. The upper right-hand image is showing the brain being placed within that tissue matrix, and you can see the infratentorial tissues, the brainstem and the cerebellum, have been removed. The bottom left-hand image is showing that same brain after it's been cut. We take a 10-inch brain cutting knife and run it through each of those gaps to create uniform tissue sections. And importantly, shown in the bottom right-hand image, this tissue matrix, upon completion of sampling, can be placed within our storage containers, which we fill with a solution of PBS and azide for long-term preservation and storage of this tissue. This is an image of all the brain tissue slabs generated from our matrix. And you can see all of these slabs are of the proper orientation and thickness. We can then use a guide and atlases to direct sampling of the brain tissue regions needed to perform our neuropathologic evaluation. The next application I'm going to discuss is how 3D modeling can be useful in neuropathology core research. A simple approach to model analysis is using defined masks. So here we've outlined different areas of the cerebrum as well as the cerebellum and brainstem. And we can use these masks to measure the surface area of each of these different regions and compare it to the full surface area of the entire model. This is a way of looking at patterns of atrophy and neuronal loss relative to different areas of the brain and to different brain models. Another slightly more sophisticated approach would be to further incorporate mean curvature analysis, where we can look at the curvature of our model to determine how there may be increased sulcal enlargement and gyral shrinking within each of our individual masked areas. And this will allow us a way to quantitatively assess patterns of brain atrophy. There are a number of other approaches as well that could be used for 3D image analysis. I'm showing two options here, one of them being MATLAB, shown on the left, and the other being Scikit, a Python-based tool on the right. Both of these systems have been used for 3D image analysis in the biomedical space before, MATLAB commonly used for neuroimages, and Scikit being used for image analysis in microscopy. And both techniques have a little bit more extended capabilities that could further incorporate machine learning based approaches to identify different mesh segments and perform different types of analyses. The last application I'd like to talk about is how 3D models can be useful for the purpose of education. When you integrate these different resources together, conceptually, you could have a space where you'd be able to manipulate the three-dimensional brain, go through the different levels of the stacked slabs cut from the tissue matrix to select a specific region of interest, say the hippocampus, and examine at higher magnification. You can incorporate your pathology stains to show hematoxylin and eosin, tau, beta amyloid, TDP43 to select a specific pathology of interest. and then incorporating digital pathology, have the capacity to zoom in even further and examine the brain at the cellular level, moving throughout the different regions of the brain, assessing different types of cells or different types of pathologies, and perhaps even able to annotate the brain in real time to do different analyses in an immersive space. In conclusion, I believe 3D modeling has important benefits to neuropathology core practice, research, and education. Regarding practice, 3D printed tissue matrices allow for precise brain cutting with control over brain orientation and section thickness. And while not discussed in this presentation, doing so also enables co-registration with postmortem ex vivo MR imaging. 3D modeling also has interesting applications for research. We can begin to think of atrophy beyond the mild, moderate, severe, and start to think of ways to have quantitative metrics of structural atrophy using different approaches like 3D mesh segmentation and analysis. Lastly, 3D modeling has important benefits to education of the brain. By integrating neuroimaging and digital pathology into this 3D modeling system, we can conceive of an immersive, augmented, or virtual reality environments, which would allow a student or a trainee to engage with the brain in a unique way and also have a distinct appreciation for the healthy brain as well as the changes that come of neurodegenerative diseases. I would like to thank you for your time and attention. If you have any more questions or would like to discuss this further, my email is up there in the top right corner. I'd also like to acknowledge my laboratory, which has helped support different aspects of this project, particularly Savannah and Mallory. Savannah is a research associate who did a lot of our model generation and 3D printing, and Mallory is a histotechnician who did a lot of the 3D brain scanning, as well as sectioning and staining of the tissue. Honor and Caitlin are both graduate students in my lab that have worked in this project in different degrees. And then Amanda Watson is our brain bank coordinator who helped enroll this particular case that was shown today, as well as many other cases in our brain bank. I'd also like to acknowledge my funding with a particular thanks to the National Institute on Aging for our Alzheimer's Disease Research Center grant. Again, thank you for your time and have a great rest of your day.
Any questions?
Hi, Kevin. Chad Murchison from UAB. This is super exciting, in my opinion. And one of the most intriguing things that I saw is when you did the overlays with digital pathology. Do you think there are any opportunities in the future to really kind of take this next step where we kind of integrate beyond like very rudimentary cell counts or area-based calculations to integrate into regions of interest and actually get like real volume metrics to really get more refined quantifications for what kind of pathology burdens that we get in the future?
Yeah, no, absolutely. And I think there's an RFA right now looking specifically at digital pathology. And I think a lot of different brain banks are having a different approach. I think the tough thing with digital pathology is all of our different centers used targeted sampling and assessment of different brain regions, right? It's impossible to take every region of every brain and get stains on them and to get that digitized, but I think even with our targeted selective sampling, thinking about organizing that data in a three-dimensional space, again, to me, really would lend itself incredibly well for just basic assessment of tissue, but also really cool analysis, considering, yes, the, structural component of, selective vulnerability in neurodegeneration.
Great. Really looking forward to seeing where this goes. So.
Awesome. Thank you.
Hello, Dr. Biniak. This is Dr. Brittany Duggar from UC Davis.
I recognize that voice.
Fantastic, phenomenal, very immersive, dimensional talk. Wanted to make sure to state that. So when you think about the workflow You said 36 hours for one print. So if I wanted to set this up in my lab, I'm being selfish because this looks freaking awesome. I even want to maybe take it to an elementary school classroom. That would be cool too.
Yeah.
So how long is that entire workflow to your end product? Like, because you're dealing with the brain, a fixed brain, right? Then you're printing, then you're scanning, then you're doing all that kind of stuff. How long does that take? And the cost too, that would be good.
No, exactly. So 3 great questions. Workflow takes about 30 minutes to an hour to scan, 30 minutes to an hour for model design. The printing is the longest at 36 hours. We've tried a lot of different things, dissolvable filaments, polyvinyl acid filaments. We just do a basic PLA. It's very cheap. These brain boxes cost, $5 to $10 a case to build. And it's very low infill within that space as well. In terms of, scalability, I think on a per case basis, $5 to $10 isn't much to do. But yeah, really, if you want to do this on a scaled up basis, you probably need 2 or 3 printers, and those alone will probably run anywhere from, the Raise 3D is about $4,000. So I think the hardware is kind of the biggest thing. And once you have that, the workflow timeline isn't, incredibly burdensome in terms of scanning for an hour, making sure everything aligns, and then creating your models.
Thank you, Dr. Benchek. This concludes our first section of the Data Core pre-meeting workshop. I'd like to thank all the speakers, and although we had some little tiny technical issues with this hybrid format, and this, by the way, is our first in-person after a pre-meeting workshop. We didn't even have the hybrid ones after like 3 years. So again, thanks to all the speakers. And it was really great to see the diversity of representation from the different ADRCs and different expertise, all the way from informaticians, data managers, statisticians, epidemiologists, computer scientists, and neuropathologists in the mix there. We had such a wide range of topics. And again, apologies for not being able to get to every question, but think of this as like as a starting point, as a way to like spark this conversation, and let's continue this throughout this Fall ADRC meeting and beyond. And with this, I would also like to thank my colleague Yorgos Tripodis for running around with his laptop so that all the speakers can do the Q&A. And this is the time that we're taking a 15-minute break. We're running late, apologies for that, and we'll resume this session where we're going to be doing our overview of the electronic data capture overview, work group overview. So I hope you all stick around.
Okay. All right, I'm going to go ahead and get started. Hi, everyone. It's nice to see so many of you in person, and it's nice to have so many others joining us virtually. I'm Sarah Biber, and I'm the program director at NACC. And I'm excited to welcome all of you to the electronic data capture update portion of the workshop today. There has been so much exciting work that this group has been doing, and we have some really, really great progress to share with you. So welcome. Going on to the next slide. So there is, I want to sort of set the context for the updates that we're going to share today. So there's a huge amount of modernization that is happening at NACC right now. And the work that's happening as part of this electronic data capture work group that you're going to hear about today is a huge, huge piece of that. And so I'm not going to go through all of the pieces of this figure here today. I'll be talking a lot more about this tomorrow during the NACC session. But just to say that we are developing a modern submission interface, which you'll hear about, which has largely been developed in collaboration with our electronic data capture work group. We're moving all of the NACC data into the cloud. It's all going to be organized using a platform called Flywheel, which is going to integrate our existing data streams, all sorts of new data streams from the ADRCs, including bringing in various types of metadata analysis data from our partners at SCAN, NCCRAD, and NIGADS. We're going to be making all of that available and searchable through a one-stop shop data search interface we're calling the data front door. So all of this modernization that is happening is happening alongside this transition to UDS v4 and is really all aimed at bringing us in alignment with NIH's data science strategy. So there are, and all this work is, right, we don't want to build it and hope that people come. That is kind of the theme that goes along with everything that we're doing and building at NACC. We're trying to do all of these builds in collaboration with the community, the ADRC community, with researchers, getting input, co-developing solutions as much as we can. And I think this electronic data capture work group and the way that this is played out really epitomizes that spirit of work and will be critical, to the success of this and really making sure that we're building something that's going to be of value to the ADRC program and to researchers. So today I'm going to talk, of course, about the electronic data capture workgroup piece of this and how we are working to build a streamlined submission system. Just a little bit of background here. So this was really in need of an update. The existing system is 20 years old and really needed to be updated to be more cloud friendly, to be virtualized. So the goals here are really to better serve the ADRC program. So we want to streamline UDS collection. And we want to be able to continue to host a range of options. We also started with REDCap because REDCap is the canonical tool for forms data capture. In addition, we surveyed all of the centers. This was back last January, and either 20 centers were already using REDCap, and 9 were interested in using REDCap, and 8 were not interested. That was before we really tried to convince them. So I think that number may go up. So this is how we will be collecting data from ADRCs using this modern electronic data submission interface. So in the future, all data will come into NACC all forms data will come to NACC via REDCap, either by direct data entry into NACC REDCap, by synchronization with local ADRC REDCap instances through REDCap APIs, or through bulk upload of CSV files into NACC REDCap. We will continue to host PDFs for the time being for UDS v4, but more and more of this will be coming into NACC via REDCap. And of course, one of the big advantages here to doing it this way is that forms can then be used by researchers, clinicians anywhere in the world on any device, amongst other things. So that's another advantage. So this group is pretty new. It was launched last January in collaboration with the Data Core Steering Committee. We've had 75 people across 29 different ADRCs that are collaborating across the 3 different areas of work, which include defining requirements, developing forms, co-developing forms in REDCap, and then developing documentation and training materials. And so we calculated roughly, I think it's more than this, 1,500 people hours that have been donated by people across the community to make this work. And it has paid off because we've made enormous progress. So I want to just share, folks here today will be sharing some updates on the progress that has been made, but I want to give just a high level overview. So for our development work group or subgroup, they have streamlined and standardized the REDCap submission process, put quality checks and variable names in place. They've established the infrastructure for longitudinal data collection. A requirements subgroup has determined the common UDS data flow used across 29 participating ADRCs. We will be surveying all 37 ADRCs on this in the near future, but these are the 29 that are currently actively involved with our EDC work group. They've also developed a data management plan that is in alignment with NIH's data sharing policy. And that is something that we'll be providing to the ADRC community. So people can take that and use that as part of their renewals when they're submitting those. We've also summarized the data quality checks for UDS v4, and these will also be shared with ADRCs so they can have good transparency in how those quality checks are being done. In addition, the documentation and training group is off doing all sorts of great stuff as well. So they've built a curated directory of viable REDCap resources. We don't want to build things that are already out there, right? We want to see what's out there and what gaps we need to fill and really curate those and make them easily accessible. and also has developed NACC-specific REDCap SOPs to facilitate ADRCs submitting UDSV4 data to NACC. We are collaborating with the EDC workgroup to develop a web page that will make all of these resources available to the ADRCs. So major progress has happened since last January, due in large part to the efforts of many of the people in this room and that are joining us virtually. So I want to thank everyone who's been involved in this effort. It's been a huge effort here. You see all the different ADRCs that are represented on this work group. There's a number who the co-leads are for these various work groups, including several members of the NACC team, Ben Keller, Carrie Stevens, as well as Laura McLeod. So you'll hear from some of those folks today, but I wanted to just say thank you. We will be having a social this evening. All of you are invited. Stop by and say hello. This is to celebrate the accomplishments of the EDC workgroup to date and really honor the contributions that everybody has made. So you're welcome to join us. It's going to be tonight, 7:30 to 9 p.m. at the Swiss Hotel in the Matterhorn room on the 42nd floor. So feel free to join us. And with that, you're going to be hearing from some other folks. I will pass off to Deborah.
Thank you so much. Okay, so I'm going to be giving an update on the development subgroup. I'm from Northwestern. I'm the data manager for the Northwestern ADRC. So the development subgroup is co-led by John Reeder from the University of Michigan and Ben Keller from NACC. And we have 29 team members from 17 ADRCs. So the purpose of the UDS4 subgroup is to design, build, and test REDCap forms that the clinical task force are developing and to build the REDCap structure and architecture that those forms will live in. So our accomplishments to date have been that development of that structure. So we have a REDCap architecture that is streamlined for longitudinal data collection and the submission process to NACC, streamlining that process. And we have also made progress in developing UDS4 forms that the clinical task force have developed and created, and developing those forms in a way that utilizes all of the features that REDCap has to offer. So we are focused on building a fully functional REDCap database. This means that ADRCs will be able to go to the NACC website and download the REDCap project. And this will be, they are ready to use the project and enter data without any additional programming on the center's part. And also for centers that are not planning on using REDCap and using another software, NACC will have the REDCap data dictionary in a CSV version on the NACC website, which will just give a big head start in programming any other database in another software. So NACC is moving towards REDCap because of all of the many benefits of REDCap. So it's highly customizable. So even though we're planning on building this out of the box approach that centers can use immediately, it's also highly customizable so that centers can make changes that are needed. This includes adding additional forms or making small changes to really customize it to the needs of the center. REDCap is also web-based and HIPAA compliant. There's also data import functions. So not all of the data needs to be entered manually. You can start with data imports as well. And the data can also be exported in a variety of formats. And REDCap also has API functionality, which just provides communication between REDCap and an external application, which allows for the creation of reproducible scripted reporting structures, analysis, communication with the NACC database, things like that. And then REDCap also has a lot of data quality features within specific forms like auto validation. So you can only input certain data types, branching logic to hide things that are not needed in the forms, and required fields to ensure that important information is being entered. So in REDCap, each participant has their own unique ID, and I'll be going over what REDCap looks like in depth in a second. So each participant has a unique record associated with their center ID, and when you go into a participant's record, you're going to see a dashboard visualization that has a lot of very important high-level information about the progress of that participant throughout their time in the study. And research study staff use this dashboard to click into different forms and use this to enter data, review data, or modify data within the REDCap environment. So this is the participant dashboard. So from this dashboard view, we have a lot of information at our disposal very quickly to just review what is going on with this sample participant. So at the top of everybody's record, you'll always have the subject ID. For this participant specifically, we can see that they have completed 3 visits over the course of 2020 to 2022 in this example. And with the REDCap dashboard, each visit in the visit data section is represented as a column. And then all NACC forms are going to be available at each visit. So depending on the packet, some forms aren't needed. So only the forms that are needed get completed. And then for this example database, it doesn't have all of the UDS forms in it. We just have a select 6 example forms for the UDS and a sample 3 example forms for the FTLD module. And you can get, again, some insights just from this dashboard, like this participant enrolled in the FTLD module in visit 2, and you know that because there is no data in the visit 1 section there. So you know this by the colors of these dots or circles on the dashboard. So gray means that the assessment or the form was not administered and there's no data in that form, and green means that it was administered and the data has been fully entered, it is ready to go, and red means that it was administered but the data entry process is still ongoing. So from this grid here, you can see that visit 1, all of the UDS forms that are needed have been completed. That one UDS form that is still gray is a telephone visit that was not needed, or a telephone form that was not needed for an in-person packet. You can also see in visit 2, again, all of the forms that are needed for visit 2 for that follow-up packet have been completed. And for visit 3, which was in this example scenario completed last week, they still have a few things to enter there, and you can see that directly from the dashboard. So apart from the visit data, we also have static data that doesn't change or changes in a way that's not associated with visits. So that is all in this first column in the dashboard here. So static data can be different IDs that are associated with the participant, an email address, other kind of demographic information that's pretty static. This also includes the milestone form. So 4 milestone stones throughout the participant's study. There could be multiple, but they don't exactly happen in accordance with each visit. So they live in a separate place, but you can still fill them out for each milestone that happens to a participant. And when and this is called a repeating instrument that you can see by the different icon next to that circle that has like a multiple kind of circles there or dots. And when there's a repeating instrument, REDCap also gives more information about how many times that form was completed. So in this example scenario, the milestone form was completed 2 times throughout this participant's journey, and and it gives you the dates as well. So that was a very quick general overview of all of the many things that you can take away just at a glance from a participant's record in REDCap. So now I'm going to move on to talk about some of the features in the individual form. So once you get away from the dashboard and within an individual form, the UDS4 will include branching logic to hide questions that aren't applicable. So you're only seeing the questions that are applicable as they come up. And for the UDS4, data will be piped from one visit to the next when applicable. So certain demographic data that only has to be entered in at the initial visit can then be piped in to subsequent visits. So that doesn't have to be entered multiple times. There will also be calculated fields that are typically used to sum up scores and score assessments, so that doesn't have to be manually calculated, and different data quality measures like ensuring data types for phone numbers, date formats, things like that, and when possible, going away from a free entry text box to something that is limiting the data entry to a specified list of response options. So I'm going to go over kind of what branching logic looks like in REDCap. So this is a A1 form that has been very much truncated to only a few pieces of demographic information. So first, I just want to point out that the date at the top of the form is utilizing one of the validation requirements from REDCap, where you can only put in a date in a specific format of month, day, year, so that all of the dates that are entered throughout this form and throughout all of the ADRC communities are all in the same format. And then for this form, the 2 fields that are going to have branching logic are there at the bottom of the form: the ethnicity question and the race question. So I'm just going to quickly fill out this form as we go. So we'll fill out the date, put in initials. We'll have this person as a research participant, and we'll have them as control. Born May 1930. And then we'll have him as a male. And then this is where the branching logic picks up. So if he does identify as Hispanic or Latino, and that is the yes is selected there, then a question that was not there before pops up to get a little bit more information about him and what region he identifies with the most. So we'll put Puerto Rico for that. And then moving on to the race question, if he doesn't identify as in any of the pre-specified options there, and you're selecting other, box pops up to add additional information. So another feature that will be utilized is piped data. So in the A1 form that we just filled out, the demographics form, like I said, that really only needs to be filled out once at their initial visit. So if we do go into the A1 form, which in this example is example form 2 in this test database. If we go into that form, we don't need to enter any more information. We've already just entered that a second ago. So when we go into this form, what you'll see is all of the data that we just put into that last form will be automatically entered into this new form in a new location, and everything will be grayed out. So it is read only and uneditable. So mistakes can't happen along the way during the study. And then a quick introduction or overview of calculated fields. We have 2 calculated fields on this example form here. One is just aggregating or summing up the semantic associations test in 3C there, and then another sum for the Northwestern anagrams test. there. So that's a very quick general overview of what UDS4 will look like in REDCap, the structure of the database, and what form features we will be using in the UDS4. So I just want to give a big thank you to the development team that has been working so hard on this. And if you would like to join the development team and join in all of the fun, you can scan this QR code or go to that link to join along. Thank you so much. And I'm gonna, yeah. And then I think the next one is.
Thank you, Debbie. Yes, it's me again. I'm sorry. For those who missed my first talk, I'm Chad Murchison. I'm with the University of Alabama at Birmingham Exploratory ADRC, and I'm going to be talking a bit about the requirements subgroup. And we're not quite as obvious what it is that we're doing. And so I do want to take a bit of time to kind of talk about who we are and what we do. I'm one of those folks who does like to lead off with acknowledgements, especially our co-leads, Carrie Stevens over at NACC, as well as Meredith over at South Texas, along with all the people who have been involved over the last several months. It has been a long, arduous, but fruitful process. In particular, I do want to make special mention of Sarah Gothard over at OHSU. I view her very much as my partner in crime on the requirements work group, and a lot of what she's done here in particular is stuff that we're going to be highlighting today. So what is the purpose of the requirements working group? Because we're not just here to tell you what to do, as much as I would love to do that. What we're really trying to do is identify and recommend the best practices for electronic or REDCap data capture in order to really kind of standardize what's being done with respect to data collection, quality assurance and control, any sort of validation steps, as well as eventual transformation as we transition data from being collected at the ADRCs to the eventual upload of NACC. So again, like I said, we're not telling you what to do. We're giving you recommendations. We're giving you best practice opportunities to really kind of make your data do the best that it can. And this is across the entire spectrum here, whether we're really kind of thinking about the life cycle of the data, from collection at the subject to the eventual upload to NACC, where it can actually be utilized in a meaningful fashion. So we also want to provide data engagement guidance and resources, whether you are a new ADRC or you're actually up for renewal. Like, we think we have opportunities to really kind of address all of these. And again, Sarah mentioned this, Deborah mentioned this, I want to reiterate this. We are recommending REDCap. It is not required. A lot of what we are providing is going to be applicable for you, whether you use REDCap or some other alternative system. So our big goals here for requirements in particular are to really kind of open up the black box of UDS and NACC data, because it can be a little obtuse at times for anyone who's really kind of worked with it. And we're trying to make it much more transparent. And by extension, we really want to promote harmonization and standardization of how both the centers and the network as a whole are really kind of engaging with data as we work to UDS4. So the primary objectives that we have, like I mentioned, are really to increase transparency of the NACC data process and just what is being done on the back end behind the curtain for all of this data that we're collecting and sending off. We're trying to kind of formalize the guiding principles of data management that are being done for the ADRCs. But again, we're trying to do this in a method that's agnostic to your site's method of data capture. You do what works best for you. And then the final thing is, we want to accommodate as many people as we can, but we do need to recognize there needs to be a starting point. And we are trying to establish the minimum REDCap requirements that are going to be necessary for sufficient electronic data capture, in particular for a site that is beginning to use REDCap for the first time. And the two things that I really want to touch on here, first of all, is a data flow diagram that we've developed within the work group to describe kind of, again, that life cycle of UDS data in terms of its collection, its processing, the way that it could possibly be asynchronous at times, and eventually leading to that upload to NACC. And then finally, this is something that Sarah had also mentioned,
We all know that January 25th, 2023 is going to be here very, very, very soon. And at that point, we all have expectations for what needs to be done for grant submissions with respect to data sharing plans. And this is no different for ADRCs. So for the visualization flow of UDS data, there are a couple of things that we really want to try to show. We wanted to make sure that we were encompassing the full data capture lifecycle for UDS data within a site. And again, this starts from enrollment of the participant all the way through to the actual NACC upload of the data, so for later utilization. We want to make sure that this flow is going to be able to accommodate both non-electronic and non-REDCap data capture processes. We really wanted to try to add clarity to the QA/QC process during uploads, and we're trying to really kind of take those next steps, which is where that survey that many of your data managers have received really kind of comes into play, where we can kind of expand things to do things like better detail onboarding electronic data capture for a nascent ADRC. So this is the diagram itself. Many of you have seen it, but for those who haven't, I just want to highlight a couple of key points. Again, we want to really kind of encompass the entire data capture system lifecycle from pre-visit data through the actual visit itself. Once the participant leaves the site, we know the data isn't done. There's additional prep work that needs to lead into things like consensus diagnoses, the finalization of the D1 form, and then finally that data upload site. And again, I do also mention doing things like better detailing onboarding is something that we're currently investigating. We also want to accommodate different capture alternatives. We can see up here at the top that we've really kind of made a point to explicitly detail a site that really wants to focus on paper collection. This is really their bread and butter. That's what they want to use because it's worked really well for them for the last 35 years. Who are we to tell them differently? So maybe that does involve things like direct data entry into the NACC data portal, using what will eventually be these REDCap forms instead. But we do want to make sure that this has its home in the diagram as well. And again, the goal here is harmonization between the sites and between NACC as just a network as a whole, and really kind of make sure that with respect to validation of the data and QA/QC, it's really kind of in place and doing the best that it can to be in agreement between what's expected at the site and what's anticipated for upload at NACC. And then the final thing, again, is really kind of, you know, pulling back the curtain, exposing the wizard, and kind of showing what things look like within the like spaghetti mesh of 20,000 some odd lines of SAS code that we're getting rid of in terms of actually processing the data within NACC so that it's nice and shiny and is fully standardized across several years and several sites across the country. So again, the goals here are to really get something that's going to be easily digestible for both new and current ADRCs, regardless of how they're doing their data capture. Again, increasing transparency, I can't reiterate that enough. That's really kind of our major goal here from a lot of these things. We again want to promote best practices with QA and QC. And as mentioned, we are collaborating with the documents work group to see if we can make things a little more interactive, since that is a rather busy diagram, and we do want to make it easier to work with. What I would argue is actually even more important is the data sharing plan that we have crafted, which is being explicitly designed for submission for NIH. And again, I do want to make mention of Sarah Gothard over at OHSU, who has done a lot of work in drafting this all together. So as we know, these submissions are eventually going to be expected. There's a 2-pager that's going to be reviewed by every program officer, regardless of funding award or opportunity. And what we've developed here within requirements is to get something that can do just that. It's been scoped to address the 6 elements required by NIH, as you can see over there on the right. It is specific to what's being required by NACC, but we're also accommodating any of that locally collected data, very similar to the stuff that I was describing in my earlier presentation. And again, this can be used by any applying ADRC, whether they're new or under renewal. With respect to data types, these are going to be things like the 5 types of NACC data that we're all used to collecting: UDS, neuroimaging, pathology, biological samples like plasma and serum, as well as genomic data. However, we're also making sure that there are opportunities for the expanded data types for NACC or the centers, because there's a lot of exciting opportunities on the horizon, and we want to make sure that there's room for those as well. We've added language for the appropriate metadata that is describing the data, because that is required. And then we also, again, allow for center-specific data collection and protocols that can also be associated with projected recruitment numbers. For related tools, software, and code, again, REDCap is preferred, but it's not required. But we do emphasize electronic data capture. We really do think that's the best way to go about doing this. We describe additional tools that are already in place that many of you are already using, such as the Naculator out of Florida. Again, you can also highlight local proprietary resources that you're developing that may be important for data use agreements. And we start to begin to describe the upload process using the NACC online portal. For standards, we do define access and conventions for data standards of NACC data, which is required for submission. However, there is room for common elements that are going to be unique to individual centers as you collect additional data to help support the UDS material. These materials can be provided by NACC if there are things that NACC requires, or it's things that can be brought up by the local sites. And we also include modifications that are necessary for instruments that we have adapted for ADRC purposes, a good example being the modified CDR. For preservation, access, and timelines, again, we go into further details of the upload system to NACC because that sort of data sharing is very key for what NIH wants. We talk about the concurrent data processing that's done by NACC, but also retention at the local site because we want to make sure that you have copies of your data as well. We emphasize the importance of persistence of IDs as well as that mapping to go from the center ID to the NACC ID and back again. And we start to talk about the NACC data request process. For the distribution and reuse, we continue to detail that data request process, start to roll in considerations for protections of human subject data. We also talk about site-specific things that may actually have their own institutional regulations, and you may not actually want to share or be in a position to share. And we want to make sure that those things are being acknowledged. You don't have to share, you should, but you may not be allowed to, and this does need to be mentioned. And then finally, we start to describe the organization and good clinical data management practices of the upload and storage systems, again, used by NACC. For the oversight, we again emphasize the delegation of regulatory control. NACC data is subject to NACC oversight. Center-specific data is regulated locally. We have more information on how the portals are being used for uploads and access, as well as the data summarization reports that are provided by NACC on a regular basis. So where do we plan to go from here? We have already sent out the survey that Sarah has mentioned. Most of you responded. It has actually been very, very informative. And so thank you for doing that. If you haven't, please do so. We look to take that 2-pager and expand that into a guidebook and roadmap of recommended practices that sites can use to build their own DMPs to actually have on site for their records. The NIH 2-pager is currently being reviewed by Nina and Cerise over at NIA, and their feedback is going to be very instrumental as this reaches its final form. And then, as mentioned, we're helping curate these lists of data errors and warnings that are already in use by NACC to better, again, harmonize QA/QC across the centers and with NACC as a whole. That being said, as Debbie mentioned, we're the ones driving this. We would love to have more help. It's been a lot of hours so far, and there are many more in the future. And if you think you have a role here, we can definitely find one for you. That will not be difficult. So please go to the survey link. We have the survey code here. Use the QR code. But again, thank you. And again, your feedback is valuable. If you have not yet completed the survey, or as you get surveys in the future, please take the time to fill them out. That's what's going to drive this process and make it the best it can be. Thank you.
Hello, I'm Laura McLeod. I am the Senior REDCap Administrator with NACC. And I'm happy to be here with you today. I'm going to talk about our documentation and training subgroup. I am the co-lead of this group with Sarah Gothard and Leah Ryder. And we have all these wonderful ADRCs who are participating in the work group as well. So the purpose of our group is to create and consolidate electronic data capture materials and resources for ADRCs to leverage in collecting and uploading NACC data. Our accomplishments to date are that we have consolidated and enumerated some pre-existing REDCap resources. We are in the process of developing NACC-specific REDCap SOPs that will facilitate UDSV4 data submission to NACC. And we are collaborating with NACC on designing and developing a web page to make training resources readily available to the ADRC community. So our end vision is the consolidation of resources on the NACC website that is intuitive and highly accessible. The website will include descriptions and definitions of NACC-specific REDCap development, REDCap and NACC SOPs, and materials from the other working groups. So what you're seeing on the right-hand side of your screen is just a mock-up of what the website might look like. It's just meant to show that we have headers and subheaders, folders if you wish, where folks can find the documentation that they need. We do want to put folks on alert that as we develop SOPs, they're going to be distributed to members of the full working group for review and feedback. We look forward to hearing your thoughts. And just like to acknowledge and thank all of the members of our subgroup listed here. And I think that we are ready to take questions at this point. Again, this is the survey. I think Debbie and Chad have done a wonderful job. I was checking my email really quickly before I came up here, and somebody's already signed up. So kudos. And yeah, we're happy to take any questions, and they can be directed to any one of the subgroups.
Does anybody have a question?
I'll start with the hardest question. But first, I want to say congratulations to everybody that's been involved. It's tremendous effort, and I think you've accomplished a lot. Sarah, at the beginning, what? Oh, thank you. Steve Edlund, data core leader at UC San Diego. Thanks. Yeah, again, amazing amount of work. Sarah Biber, when she presented, was very careful in the way she spoke about HIPAA compliance. REDCap is not HIPAA compliant, but it can be made HIPAA compliant. So maybe just help us anticipate the steps and, you know, assuming that the NACC cloud is HIPAA compliant, how much of that do we inherit from NACC, or do each of the institutions have to? I'm just again, this is probably the hardest question first, but I'm wondering if you could help us anticipate that.
Sure. So any nonprofit is able to license REDCap for free. And so there's over 6,000 organizations globally who license it. And it is up to each licensee, each holder of the REDCap license, to maintain their instance in a HIPAA compliant environment. So if an ADRC has their own REDCap instance and they're collecting data on that REDCap instance, then it's up to them to make sure that their environment for their instance is HIPAA compliant. I do believe we are going to offer an option for ADRCs to use NACC's REDCap instance, which is maintained in a HIPAA compliant environment. Does that answer the question?
So I want to follow Stephen's question. First of all, again, thank you all for such great work to do this. It's really helpful to all the ADRCs, I would say. One question I have about is the Part 11 compliance. This is really important in clinical trials, especially the regulated trials. So I think, Debbie, you made a comment, this will be Part 11 compliant. I think you're talking about the Part 11, the REDCap cloud. The system is Part 11 compliant, but is there a plan to actually make the entire NACC UDS database eventually Part 11 compliant?
That's a really good question. It is a very complicated and lengthy process to make a REDCap instance fully fully compliant with Part 11. There are some institutions who have done that. There are components within REDCap that are Part 11 compliant, including the e-consent signature part of it. But that's something really good to consider, and we can certainly look more into that. I don't know about NACC's future plans for that. And then, of course, any ADRC that is hosting their own REDCap instance, it is very possible that they may be Part 11 compliant.
Great, thank you.
I saw Sean's hand up in the back.
Hi, Sean Mooney, University of Washington. Just a couple of quick comments about the last two questions. You are, I think someone mentioned this, you're correct. I think REDCap can be HIPAA compliant and it can be Part 11B compliant. And there are costs and kind of things that have to be done outside of REDCap in order to make that happen. when it comes to HIPAA and research, things get pretty complicated quickly because some of what you have to deal with relies on your institution that the ADRC is sitting in and what their policy is around what data needs to be HIPAA-like and what data isn't HIPAA-like. And so I think, it's worthwhile for each ADRC to talk, to understand what their institutional policies are in that space. We can put controls and, and configure REDCap to be very HIPAA-like from a security perspective, but in terms of a policy, compliance regulatory perspective, that's really very much an institutional specific issue. I'd be happy to talk to anybody about that more. Laura is maybe underselling herself a little bit. She sits along, collaborates very closely with the rest of the University of Washington's REDCap team, which manages a very, very large, also HIPAA compliant REDCap instance that actually does have operational HIPAA data in it.
Oh, goodness. I'm Jess from the University of New Mexico. I just barely got involved with the ADRC that's still fairly new there. So forgive me, first of all, for missing all the earlier meetings. We got here late. I just was wondering, do you have a standardized procedure for like QA and QC? Do you use like the REDCap QC process, or do you have each center develop their own QC process?
So that's a great question. Right now, it is the Wild West in terms of kind of what's being done. And we are trying to make that not the reality. One of the things that I have learned in talking with other sites is the amount of reliance that's being done on QA QC solely by NACC at this point. And that's not really the way it should be. And I envision like this wonderful golden utopia where I run my data checks and I upload it to NACC and it works the first time. And my God, it's glorious. Like, I absolutely love it. And like that, and that's what we would love to be able to do. But we know that is a challenge. And what we're really trying to do at this point is develop the tools that are going to be necessary to help inform sites. This is what you expect, because right now you can actually go to nacdata.org and you can download the collection of QA/QC rules that are being done through the legacy SAS code that NACC has been using for 20 plus years. And if you print it out, don't drop it because it will break your foot. It's so thick. And so we're trying to find better ways to let sites know this is what is being expected to get a clean packet and put that into place. And REDCap can accommodate a lot of these quality checks. You would be amazed how much they can actually do. It cannot do all of it, but it can do a lot of it. And we do acknowledge that some stuff needs to be done. But what we really want to do is encourage sites to do their own QA/QC, if for no other reason. It's just best practice to do that. It's good clinical data management to do it at your site because you know it's good at that point, because that way other collaborative sites can approach you directly and get not only your UDS data, but other data you've scrubbed under a similar diagram and be able to share that. So that's what we'd really like to do. And there are better ways to do that than, the 150-page PDF. And that's what we're trying to work towards, but it's going to be slow and it's going to involve a lot of conversations with other sites. What are you already doing? What are you relying on NACC to do? What can we do to empower you? And that's kind of where things are at this point. So.
Thank you. Nearly from the UTRGV South Texas Alzheimer's Center. I would like to know, are you envisioning to implement this directly with the UDS4? What's the time frame to start implementing this? And also the other, I know it's a hard question, but the other thing will be, is this going to be directly, the data should be uploaded directly from the operator, from the the clinician doing the interview directly to the REDCap? That would be very interesting. So if we can use a format on a tablet or a mobile device that is also implemented in REDCap, is it going to be friendly for that, that kind of input?
I don't want to speak for Sarah because she can answer that first part far better than I could. What I will say is that we are beholden to the clinical task force and their approval of the forms, and that is an ongoing conversation. So I'm not going to give any estimate, and I'm sure that Sarah will have much better details about this tomorrow. For your second point, one of the things that we're actually actively querying from the sites is, what are your methods of data collection? How much paper form do you do? Who is doing your data entry, whether it's the data core, the clinician, whomever? Are you interested in direct data upload? Are you interested in direct data entry? And by that, we think about things like the mobile tablets for surveys and things like that. UAB, for example, we send out the B8, B9 for the clinicians using REDCap as a survey. It's directly entered. They don't bother with paper forms, but we're also beholden to have a very minimum paper footprint in this regard. So we are keeping this in mind as we move forward so they can be implemented. What we're also trying to do is make it a starting point. So you can actually take these designs within REDCap and build out a prettier survey using things like field embedding, and I don't want to get into the technical details, but we're trying to put you as a site in a position to mature the forms to something that's going to be very tailored and suitable for you in particular. So we are keeping that in mind, and we want to find out how much interest there is by sites in order to do this, because that tells us where we want to direct those resources and as we move forward. So
thank you.
All right, if we don't have any more questions, I'd like to close this session, and thank you everyone for talking about the EDC working group. And among all the incredible 1,200 hours of work they have put in and all the output, they've also brought us back on track. Instead of being 20 minutes late, we are actually only 5 minutes late. So thank you for that, for giving us. So again, someone like Jess, you, from Mexico, please, think about joining the EDC working group. It's a very open working group. We have 79 people so far, or 79. So yes, we would love to make it 80. So please think about joining that group. And they have made it really easy. It's a little QR code that you can scan on your phone and join these meetings to learn more. Thank you, everyone, and see you all at the ADRC meeting.